day of this webinar on nanotechnology. I'm sure you had excellent deliberation yesterday with the eminent speakers. Today we are having 4 lectures focusing on nanomedicine, nanomaterial in plant protection, nano food technology, and nano bioremediation. Today's first speaker is Dr. Jagat Rakesh Kanwar from AIMS Bhopal. I feel privileged to just read his brief biography. He earned his doctorate from PGIMR MER Chandigarh. Before joining as head department of biochemistry AIMS Bhopal, Dr. Kanwar, professor of nanomedicine group and group leader of nanomedicine laboratory of immunology and molecular biomedical research, School of Medicine, Deakin University, Australia. He is highly cited researcher and he was globally top among 2% highly cited researchers around the globe in 3 disciplines of the science, field of oncology, medicine, chemistry, and nanotechnology. Dr. Jagat Kanwar is nominated member of Association of Universities of Asia and Pacific Research and Innovation Committee. He is recipient of Vice Chancellor Prize and Award, Vice Chancellor Award for Excellence in Research, Deakin University. He has national and international reputation in investigating the fundamental and applied molecular aspect of cancer and chronic inflammation. His research is also focused on miRNA, log nucleic acid, LNA modified chimeric aptamerase siRNA conjugates, and working on nanotheranostics and nanonutraceuticals, nanotechnology, nanomedicine based protein and peptides. His lab has discovered novel and safe targeted nanomedicine based on nanonutraceuticals for cancer, autoimmune disorders, and inflammatory disorders. Professor Kanwar's publications are there in international and peer-reviewed journals in the field and have added the body of knowledge in the field of cancer therapy, cell biology, immunology, nanomedicine, gene therapy, and nanobiotechnology. His research articles are cited in more than 9,000 times by the scientists with HI index of 55 and I-10 index of 160. So, with this eminent background and such an excellent researcher, we are privileged enough to have and listen him in this webinar. Now, I welcome Dr. Kanwar to deliver his talk. Sir, please.
Thank you, sir, for introducing, and thanks for inviting me for this excited meeting. I learned a lot, and as mentioned, we did a lot of work on nanonutraceutical use of different material from natural resources and use for. Can you see a slide?
Yeah, you can see, sir.
Yes, sir. It's visible.
It's visible. Okay, so I'll make it full screen. I don't know why it go to the last slide. So this is all about the nanomedicine. So thanks for a nice introduction and inviting me for this excited webinar. And thanks especially Dr. Anil Kumar for inviting me and having interaction few weeks ago in our institute here in AIMS Bhopal. So as mentioned, I'm professor and head in the Department of Biochemistry. I'm basically from Himachal, and my matriculation from Himachal, very remote areas. Then after that, I did my graduation from DAV Jalandhar, and then master from PAU Ludhiana from Department of Biochemistry. I have a direct relationship, or you can say many interaction with agriculture scientists, soil sciences, pathology people, veterinary sciences in PA Ludhiana. After that, I did my PhD from PGI MER Chandigarh, and this, as you know, is a very renowned institute, and it's also second in the medical sciences and publication in, and I think in whole world, that's in 15th in position after after AIMS New Delhi. So we learn a lot. After that, I moved to Auckland University. I worked there for 10 years in School of Medicine. 15 years I'm working in School of Medicine in Deakin University. Is situated in Victoria. So I introduce you a little bit of where I. So first of all, I think I go other way around. I'll acknowledge because most of the time when I give a talk, sometimes we miss the acknowledging the people who contributed or those who funded for this research. So first is the Auckland, where we work for 10 years. This is the Fonterra Center. Fonterra is the second largest company in the milk. Introducing because it's a direct relationship with the agriculture, so dairy sciences. So we work a lot. Each year we got a $5 million to develop the nutraceutical from milk. So we did a lot work and patented more than 30 different patents in milk product as a nutraceutical. There we had a consortia. I was the chairing this consortia. That's a Lacto-Pharma. As name indicated, lacto means milk, pharma means pharmaceutical.
Not moving, sir. Your slide.
Okay, so it's moving on.
No, sir. Please come to your first slide. Means
This is your Google Scholar page.
the Google Scholar page.
So what I do? I'll share again. What about now?
It's fine.
It's moving.
Not moving, but we are. It's moving now.
Now it's moving.
It's moving on. Yeah. So this I introduced you to my university, Deakin University. It's in Victoria. Victoria is the smallest state here, and this has three campuses: one in Melbourne and another with Geelong. Geelong campus. Can you see the icon where I'm moving? Yeah. Can you see?
Very much.
Sir, yes.
Okay, so this is the Geelong here we were situated because School of Medicine, all engineering sciences, biomedical engineering were situated over here. There is another campus. This is 300 kilometers from this campus, but the other campus in Geelong is just a 60 kilometer. Take maximum 40 minutes. There is no light in between, so it's a full driveway within 40 minutes. You reach over there. So the School of Medicine is a big School of Medicine. We have more than 150 students from all over countries. We have more than 15 seats reserved for overseas students here. Most of the time Indians, they capture these positions. We also have optometry. We also have imaging in School of Medicine. So first I'll introduce you the talk which I'll try to cover today for giving a complete view or a picture how we will see. So this is the Lacto-Pharma, which is a lactoferrin. As you know, this is produced from milk, and it's also present all secretion. Even tears also have these proteins. How this playing a role for tear when secreted from our tears? This is another big lecture. I'll give this lecture later on. I'll try to cover little things for today. So if you focus over here, there's a red dot over here. These are the iron, as Dr. Anil already introduced you. It's very important. Iron deficiency, anemia. A lot of people have a pregnant woman, and in our childhood, a lot of kids they have anemic in our country as well. So it doesn't mean you are a vegetarian or non-vegetarian. Even the people who are eating red meat, they are also deficient for this this iron. And if you have a depleted iron, then your RBC is not matured and you will not get enough oxygen transported to your body. So lactoferrin is a protein which is abundant, mostly present in a milk, especially in colostrum. And that's the reason doctors they recommend at the early ages to have a breast milk for our kids, because this has a lactoferrin. This immunomodulator. This protein has a multi-functional protein, has a anti-cancer, anti-inflammatory, antiviral, and anti-fungal activity as well. So iron play a major role. But in nature, in milk foam, we don't have more than 10% saturation of iron in this protein lactoferrin. So what we make this very unique, we make this 100% saturated with the method which we patented. So now the protein which we are using in our rest of these experiment or the study, which I'm going to show you in next few slides, is 100% saturated. 100% saturated. All the pocket of this lactoferrin has a iron. So it's a iron saturated. In addition, we also play a trick, or we develop a technology where we can use this protein as a contrast material by replacing with the contrast materials such like a superparamagnetic nanoparticle, zinc ferrite nanoparticle. Dr. Anil already introduced you the superparamagnetic Fe2O3 yesterday, why they are needed, and how we have in our body, we have a lot of metabolic role in our system. So beside this protein has a application for immunomodulation. So if you are not taking enough lactoferrin, even lactoferrin had a big role in COVID as well, because it's immunomodulatory product. We also did the clinical trial. We are using this iron saturated lactoferrin for COVID, for other application for the cancer study as well, and use as a contrast material, because most of the contrast material, if you talk to the person who did the angiography or who has been done with the angiography, the doctor might have, and the patient might have experienced the toxic issues with these contrast materials. So we try to replace these contrast materials with the natural form or modified form of these iron saturated or superparamagnetic nanoparticle based conjugated lactoferrin, so that the side effect of these contrast material get reduced or minimized or no side effect. Another protein which I want to introduce you here, the survival, as name indicated, survive. So it's allow the cell to survive for a longer period of time, and all the tumor cell, you name it, any cancer cell have overexpression of this protein. Another molecule which we did, because as you think that if the protein which is responsible for cell growth in cancer cell, you can deplete, you can make antisense, you can make dominant protein, you can make a siRNA, you can make a microRNA, and you can play around to kill the tumor or induce the cell death in even chronic stage of inflammation. So what we did over here, because when we developed this as in 2001, when this protein has been identified, first time we made an antisense or a dominant active gene therapy, but at that time gene therapy was failed due to some reason, due to some regulatory issues. So what we did after that, we made a protein. We make a more safer and protein form of this dominant active survivin, so which dimerize with the survivin protein which present in all cancers or in chronic inflammation and induce the cell death, so that cell die. Another molecule in cancer or inflammation is very important. This is BGF. This is the vascular endothelial growth factor, which present in overexpressed in all these blood vessels, which is responsible for angiogenesis or inflammation in chronic inflammation. So now there are a lot of antibody-based these VEGF is present, but again, problem is antibodies are very big. So if you give antibody, then might be some of these autoimmune responses also occur with these antibodies, and they are big. Absorption is a big issue. Clearance of the system is very big issue. So we develop aptamer. Aptamer is nothing. It's a chemical antibodies which are responsible to play better role as compared to the antibody, because they are very small in size. They absorb very fast. You can modify them. With modification, you can make the absorption of these small molecules as a aptamer better than the antibodies. But problem with these aptamers, again, they are the nucleic acid, and a lot of the restriction enzyme, they start to degrade or degenerate with the after some time. So we make a technology which I introduced in the next slide, how we did these assays. We make locked nucleic acid aptamers. So means the active site which are responsible for the restriction for the acting of the restriction enzyme. So we make it unavailable to these restriction enzymes. So we play a lot of role where we use in blood cancer and combine with the lactoferrin and with the survivin, so playing a better role or synergistic effect. Another assay we also did with locked nuclear siRNA for survivin, as Dr. Anil already introduced you yesterday, there is the SIR and a microRNA technology. So this is the advance of this technology where we use log nucleic acid to conjugate with these SIR and do nucleic acid so that restricted enzyme, they can't act on this, they can't degrade. So half-life or the life of these molecule you can prolong. Another molecule which I want to introduce here, trichostatin again, this is also natural form. They are separated from the fungus or the bacteria from these from natural resources, and they have been used for many uses. Like if you see these days people are going for refractive surgery. You don't want glasses, so people, especially the youngest, they don't want glasses, so they going for the surgery, refractive surgery. So doctor prescribe you some of these eye drops for a longer, so that you don't develop the haze, or you don't develop what you call scarring. Scarring is the effect which comes after surgery. So laser is also responsible for these scarring. Another molecule which we also identified recently is the MMC. This molecule also stops the scarring, and in eyes, scarring is very important. And you know, when our soldiers get wounded, or some have some chronic inflammation, have some allergy, chronic allergy for their eyes, or some injuries, or some get exposed in our laboratories with the acid or base, then there is an injury. And even with the laser, as I mentioned, refractive surgery, they also induce the wound, and when there is a wound, there is TGF beta induced, and TGF beta is responsible for the haze or scarring, so the vision is impaired. So this is the introduction, some of the molecule which I'll try to cover today. So now, big question in cancer is the diagnosis. There's a lot of problem with the existing chemotherapy, existing diagnostics in our cancer detection. Major issues is the early diagnosis, and most of the diagnosis so far we have, they are the invasive. So we want a test which are gone for the non-invasive technology, like a urine test by way of saliva or some other base, or with the tears. You can identify some of the markers which are responsible for the cancer. Another is the cancer cell or the cancer resistant cell and cancer stem cell. And they are the culprit. They are responsible for migrating from one part to the another part of the body. And you can imagine when somebody have a cancer resistance or the drug resistance, then these cells, they are not responding to any chemotherapy. So we don't have a long-term plan to have a individual which are cancer drug resistance. We can treat them. We don't have a individualized individualized or personalized medicine so far for the cancer, except the breast cancer. Issue of the off target. All the drugs so far we are using, they are off target. They go everywhere. All the drugs are toxic. Even the plant source Taxol, which we use for the cancer, is also toxic to normal cell. So how we can avoid these ones? So question, and the answer is the encapsulation. Dr. Anil already introduced the encapsulation. Why this one? Today I'll give you some data which we did or use for the inflammation or with the cancer. So another is the effect of the limited effectiveness of cytotoxic agent. The agent which we are using, as I also introduced the Taxol, which is also isolated from the plant, is also toxic for the normal cell. So you can avoid. So you need an answer for these the question for the future. Immunoediting, as you know, when somebody have a cancer and the cancer is big enough, then it's not treatable. The patient has to die, means one day they will die because of our immune system is not responding, and the molecule or some of the toxic chemical which are secreted from the tumors, they are inhibiting or editing our immune responses. Another factor is the monotherapy. As you know, these days when somebody has a cancer, the doctor, they prescribe more combination therapy, so not a monotherapy, because when there is a monotherapy given, even in the antibiotic, when monotherapy comes, then drug resistance comes. So doctor usually prescribe high doses, and then they go for the combination therapy. So now the answer with us for in cancer, especially in cancer, is the multi-modular. Modular means we have a more techniques. So like we have a magnet nanoparticle, we can use a magnet to scratch the tumor cell, and we can use the infrared waves, We can combine multiple therapy with radiation therapy, chemotherapy, make it more targeted, or with nanoparticle, making a multifunctional nanoparticle so that these nanoparticles that are target the tumor site and then kill the tumor much faster as compared to the monotherapy or single therapy which we are practicing in the past. So in future, there are a lot of molecule is in emerge. So this is the first slide which we did in Auckland University, and this has been patented. And now this molecule DMXA is also a part of Sigma catalog. So this is the across all the different phases, and this is the UK-based anti-soma company which are continuing this work for us, and this is also present a Sigma catalog. You can see over here results over there, and this is the gene therapy with the help of the anti-vascular therapy with DMXSA in combination with the immunogene therapy. So you say here within 29 days, whole tumor is disappeared, and here we were able to develop the systemic immunity. When we call the systemic immunity, means the immunity which we develop in a body that's present in all. So if we challenge the tumor with another type of tumors, then they will not come again. So we do on same mice, we develop a multiple tumor on same mice. One, we use the gene therapy, and then you can see within 29 days, all tumor get disappear. So this is a catalog, and this will be published in a very nice journal, Cancer Therapy in 2001. And yesterday you got introduced by the functional medical food. So this is the first medical food I'm proud to say, where we use the iron saturated lactoferrin and combine with the chemotherapy in breast cancer patient in New Zealand, where we put the iron saturated lactoferrin in a ice cream and 1 scoop a day, then disappear the effect of all these side effect with the chemotherapy which we tested in breast cancer patient. So now question comes the nanomedicine or the nanotechnology. When we started work in 2001, the term nanomedicine was not well defined, but although there are few publications coming from the nanoparticle, nanotechnology, because this has been defined in the 1960s, where first time the paper has come that the nanoparticle play a role for targeted therapy. Then FDA come up, then they only approved the PGLA nanoparticle and the iron Fe3O4 nanoparticle so far. But the other particle have some issues, and I'm going to go to these one later on in my talk. So now nanotechnology, as yesterday's talk, all talks have already convinced you that there's a big dollar for the future, and the forecast is each year more than 23% growth is happening in nanotechnology or nanomedicine. So there is a future for nanotechnology for future. So now, as a nutraceutical, as I mentioned to you, how you use a nanoparticle or how you use a material where you can save your drug of choice. Your drug could be a siRNA, could be a peptide, could be a drug which is very toxic, and a molecule which is very expensive. How you can use those one? How you can protect your molecule for a insult which caused as the low pH in stomach and high pH in intestine, and there are a lot of enzymes. So how you protect these ones? So these are the three guys who work for their PhD program and spend a lot of time to making a nanoparticle smart, which we call nanocapsules or nanocapsules for the targeted therapy. Why we call it nanocapsules? Because they are small in size and they are like a capsules, and they are less than 100 nanometer in size. And then you can load your molecule, load your drug, siRNA, a positive negative charge of your drug on these nanoparticle and make it available to the system. So here, inner core is the calcium phosphate. It's very simple. You know, calcium phosphate is part of our body and don't have any toxic fatty in our body. So we encapsulated this one with the chitosan. Chitosan, another have two type of chitosan, animal source of chitosan and the vegetable source of chitosan. Because in India, some people don't like vegetable or the animal source of these material, and they are all mucoadhesive. Mucoadhesive means they attach to the mucus in our intestine, and here they release the drug or these nanoparticles slowly. We use the alginate on top. Why we use alginate? Because in acid pH, when you see these nanoparticles going in acid pH, they get degraded. So alginate has a tendency not to allow to degrade or available these to the system directly in low pH. And when these nanoparticles reach to the intestine pH more than 6.5, they get they're dissociated, and the no nanoparticle is available along with the drug molecule which we encapsulated or loaded on calcium phosphate, even protein, peptide, siRNA, hydrophobic molecule. So you can load all these. But we published this in a Nanomedicine journal, which has a very good reputation. Another thing, yesterday you have been told that different iron absorption, different level of iron is important. As you know, when you take a lot of supplement of iron, then you get depleted with the calcium. Because in our system, in our intestine, we have a divalent metal ion. So they can't discriminate iron versus divalent manganese, magnesium, or zinc, or calcium. So if you're taking high amount of iron, then again you get deficient to the calcium. And if you're taking high level of calcium, then again you get depleted with the iron, your iron level go down. So in our system, in our diet, we have to take a balanced form of diet where we taking a right amount of these nutrient or micronutrient in our body. So here, this is the assay where we have all these particles has been depleted, and then we get absorbed to the system, but go to the blood vessels, output to the blood vessel, go target to the tumor site. Here we found the natural form, which is very interesting to know, the iron saturated lactoferrin, which play a role for down-regulating the expression of cancer-resistant stem cell. We tested these markers. So these are the markers which present on the cancer-resistant stem cell. If you see, these go down, and when they go down, they induce or they force these cancer-resistant stem cell to induce cell death. So to summarize this data, if you use the iron-saturated lactoferrin in a diet and use a nanocapsules orally, then you can treat the cancer at the advanced stage as well. Now, another question comes with the, when you use a nanoparticle, then question of nanotoxicology. Yesterday lecture, I think all lecture introduce you of nanotoxicology, but this is again another field which is very important, and that's also effect on the surface. It happened due to change in surface, solubilization, charge on the molecule, size of the molecule, chemical composition and morphology of your nanoparticles. So these have the very important check. And intracellular level or the subcellular level, they play a role at the lysosomal, autophagy, mitochondria, and uses different signaling molecules which are responsible for killing or playing a cytotoxic effect in these assays. This toxicology not only in human or not only in living creature, but also play a role in our environment. So you have to be very, very conscious of when you're developing any nanoparticle, so or any nanomaterial. You have to test, or you have to be very vigilant to see whether they have any side effect in our nature. So, for example, you have a airborne pathogen nanoparticle, even small, as yesterday mentioned, that these nanotechnology is not only in medicine or in this agriculture, this everywhere, where you think means it's everywhere. Solar panel, you take small clothes, they are also making an animal. These days, even paint is also coming from the nanoparticle. So you have to be very, very seriously when these nanoparticles you spreading or you are manufacturing, then they are coming in our ponds or in our lakes and induce a toxic effect. But luckily these days we have assays. We have different assays where you can explore these assays to find these nanotoxicology with the help of these assay, like in cellular level, any cell. These days, the technology is so advanced. The cells which we make 2D, we make a 3D culture of these. We can make a spheroid. They are organoids these days, where we make exactly the shape and the size of these organs, and you can test in all cell lines or a human cell. So Later on, we test this in zebrafish, in rodents, in later stage in the human. So this is the very important technology which recently we are also using this as a chip assay, where we use organoids from different organ and test these nanoparticles for toxicology assay. So now I introduce you the aptamer. Aptamer, I already mentioned, these are the small chemical antibodies, and they play a better role and absorb much better than the antibodies, have no side effects, and get eliminated from our system much faster as compared to the antibodies. So we did a lot of work. If you put in Google, in aptamer, we published more than 30-40 different publications about these aptamer. But as I mentioned to you in my first few slides, the aptamers, they are nucleic acid based, or RNA or DNA based, and they are unstable, and after some time they get eliminated from antibody. The half-life of these aptamers is sometime not more than 10 hours or 5 hours. So how you can make them available for the longer period of time and make it available, especially with the nanoparticle, and how you can. Hello. Okay. So we make a nucleic acid. I think there's a disturbance if somebody's speaker is on. So here with these nucleic acid we develop, where we make stable for a longer period. Why make this one? We want to make it targeted delivery. So far, the drugs which we are using, they are off target. They go everywhere. Any drug or chemo or any molecule which you are taking going everywhere. They can't discriminate whether these go especially to the tumor. So when we make a targeted nanoparticle or targeted nanocapsule, which we mentioned, here we use the super paramagnetic nanoparticle conjugated with these lactoferrin, which is a bovine lactoferrin, and then conjugated with the LNA aptamer, and these aptamer we use, this technology we use for MRI or CT scan for real-time image. So you know not to. So this is the strategy, or this is the design over here. We publish this one in very nice journal, Impact Factor, I think more than 15, and in biomaterials, where we use a calcium phosphate core. As I mentioned, this can take hydrophilic, hydrophobic. They use iron saturated supermagnetic nanoparticle with the lactoferrin conjugated on top or adsorbed on the surface of these calcium phosphate. Then outer core we developed with the chitosan, and then on top this one we conjugated with the lock nucleic acid aptamer of nucleolin and EpCAM. We use these two molecules knowingly because we know these molecules overexpressed in tumors specifically, but not overexpressed in normal cell. So, and moreover, this nucleolin and these EpCAM translocate from membrane to nucleus. So you can imagine if you have a nanoparticle tagged with this material, then you can make it available to the inside the cell where actual action happen, because the most of the action happen inside the cytoplasm, not the outside of the cytoplasm, except you want to degrade or rupture the membrane. So no, they get encapsulated. Outer core we use alginate, sodium alginate, because we want to protect from acid pH in stomach. So we give them orally to the mice, and then develop this MRI, this with the MRI. So if you see over here, nanoparticle develop on the flank of these tumors on these mice, and you see this is the untreated one, where the tumor grow rapidly and large. And when we treated with the non-targeted, it's also there, but it's diminished. And in targeted nanoparticle, it's there, but totally clear the tumor, completely clear the tumor if you see as compared to the untreated mice. So now we use these super paramagnetic saturated nanoparticle for delivery for CT scan. This is CT scan. And now we use the super paramagnetic as a thronostic. Thronostic means for delivery and for imaging as well. So now this is the whole design of this nanoparticle. Here, the targeted, we can conjugate it with Cy5. You can load multiple drugs. You name any molecule, hydrophilic, hydrophobic nanoparticle, and side by side, you inject these and at the side of the tumor, you can give these mice with the oral, and then you can image in real time of these mice. So we're developing this technology, and we wrote many grants in here in India as well to use this one for human trials. Now we also use these because the major problem when somebody have a plaque in their heart in their blood vessels as the atherosclerosis, then contrast material, as I mentioned you earlier, has a toxic effect. So we want to replace these toxic contrast material with the safer, natural, having no side effect on these. So we use paramagnetic nanoparticle. We develop another form of nanoparticle, which are zinc ferrite nanoparticle. As Dr. Anil already mentioned, zinc is metabolizable, iron is metabolized in our body, and there is no side effect of these molecules when they're degraded inside our body. So they play a role. So we make a zinc ferrite nanoparticle, which has the maximum magnetism, and we tested this one for heart. If you see over here, they present in heart. So we try to replace as a contrast material for future, which need to be done in next stage of this study, where you can use a contrast material like this one, and you can image the whole body without any side effect and get the complete MRI scan. So we tested as I mentioned, to be not only focusing one cancer, but we tested on many cancers. So this is another cancer we used. That's the prostate cancer. These are the mice which are spontaneously develop these tumor as we develop in a human being. So we tested these one, very long study. Will give you the summarize what we did over there. We conjugated our these drugs with our iron with the lactoferrin. And this is a normal study. When this is normally happen when you take any drug, then drug go inside with the PGP pump. After some time, when you're taking these drugs for a longer period of time, they pump out these drugs outside automatically. This is the defense mechanism in our body has. So in cancer, they immediately pump these drugs out. And especially when the these cells get exposed, there is overexpression of these PGP pump on the cell. However, when we make a combination or make a conjugated form of with the iron saturated lactoferrin, they diminish or they degrade these PGP pump. Along with this one, when this get encapsulated or endocytosed, this remain in the body for the longer period of time because we conjugated with lactoferrin. And lactoferrin has a high molecular weight, and it takes time to get a released drug inside the body. So slowly this drug released and inside the cytoplasm, inside the body, and they play a role for killing the tumor. So if you conjugate these molecules with the any anti-cancer or anti-inflammatory drug, you can use these or exploit this one for targeted therapy as well. We tested for retinoblastoma. Retinoblastoma, unfortunately, in India is very high. If you compare with the rest of the world, the eye cancer in India, due to many reasons, is very high. So we also we published a lot, I think more than 20 publications where we published from our grant, Indo-Australian grant. And this we did with Krishna Kumar. He's from Shankara Nitralaya in Chennai. And we published did a lot of work on retinoblastoma. So if you see here, this is the eye here, and here this is retinoblastoma inside the eye. And within few, in childhood, the incidence in India is unfortunately very high. So we work a lot. So I can't go in detail about this one. We also use these nutraceutical iron saturated lactoferrin for bacterial, antibacterial. We use for typhimurium here, salmonella typhimurium, Giardia, entamoeba, and with the malaria as well. So summarize here that the iron saturated lactoferrin not only play a role for tumor clearance, but also play a role for antibacterial or antifungal activity as well. Another study which we did with the osteoarthritis, which is another interest of the people in biotech, in agriculture biotechnology, where we can use a nutraceutical to replace the anti-inflammatory drugs which we are testing or which we are using, because all these steroidal, non-steroidal drugs have the side effect on our heart, stomach, and our kidney failure. Most of the time, those who are taking long-term for these anti-inflammatory drugs or non-steroidal have a lot of side effects. And this happens when some of the bones, especially the elderly ages, or when somebody have a fracture, then this imbalance happen, and this imbalance is responsible for osteoarthritis and for the rheumatoid arthritis and the bone absorption. So I summarized a little bit about this, but I was invited in a lecture in Palampur. There is a one Ayurvedic University, in Palampur. So I was giving a lecture, and at that lecture I met with Dr. Sanjeev Sharma and with the Ramesh Arya. I think people from there may be aware from the Ayush point of view. And now I think he's in Jaipur, a director of Ayush in Jaipur. We were giving a lecture, and he said we have a product, and that product is very much useful, and you can use this one for your arthritis study. So one of my students from Sri Lanka, she was using a model of arthritis, rheumatoid arthritis. We tested this one, and this herbal product, and this is a combination herbal product known as a Google form, and they are in use, and they also tested during in a viral infection as well. I think they put a glowy and multiple different herbal product in this one, but here we tested these one for a rheumatoid arthritis model in our study. So this is the PhD student and the later on postdoc in my lab from Sri Lanka. So use this one for herbal study. These are nanomolecular characterization. I think it's a very long study that has been done, but I summarize you what happened over here. Then we use these iron saturated lactoferrin in nano capsules and treated these mice with the rheumatoid arthritis or arthritis. They deplete some of the acids, and here they target the inflammation and the cartilage degradation. So summarize the natural component which we tested, and as we know, they have a less regulatory regulation as compared to the other chemotherapy. So you can use these on a test for various applications for various inflammatory or chronic inflammation. We also tested for candida, antifungal. A lot of work we tested. We make another form of these nanoparticle in combination. And then we also use the neem protein for anti-cancer activity, and neem protein and the neem oil. So which we use and also we use for the replace the toxic pesticide. So to summarize this one, I was giving a lecture in a PG at Chandigarh. There is one girl, she was interested to apply for the international fellowship. So we developed a small project over there. And ICAR funded this project. So she worked with me in Australia, and we developed a lot of study, and we used a biopesticide to replace the toxic pesticides. So I'll show you some slides over there. Here, we developed the guar gum. Guar gum is again agriculture. Here we use a lactoferrin, and then you can see on these insect, they present on their antenna. So this shows that if you make a nanoparticle and then you can be available for biopesticide with the neem oil. So we use a neem oil and also use the neem leaf glycoprotein for using this nanopesticide from different. So these are the insect over there. So this is the untreated. If you see, they are happy. So when we treated oil form of this control, they are not happy, shrinking, and they are not producing any offspring for the future. So if you use a crude neem, again with the nanoparticle, you can see they die between few days, and then neem oil is much better than neem leaf glycoprotein. I think can I take 5 minutes more for summarizing my talk, or is it okay now?
Please carry on.
Okay. Another thing, what happening in our life, as you know, the eye drops has a big issue, and we don't have any formulation. So it's my call from agriculture people who are working on different herbs. They also try to develop some nano formulation, which also play role for some horror. Because what's happening when we're taking eye drop, have any inflammation or any infection, doctor give you eye drop. But problem with the eye drops, only 5% of the drug get absorbed inside inside the rest, 90% get wasted. So moreover, when there is any issue with the issue with the any drug, so they poke with these one syringe with long syringes. So we don't have any nanoparticle, any delivery system for our eye delivery system. And this is the work which we did in glaucoma. And as I mentioned, 5% is get absorbed, other get wasted. And in eye, because eye is situated in an area where only 0.02% of the drug which giving orally goes to the our eye. So if you increase the dose, then you increase the side effect of the drug on the body as well. There are few these intraocular implant are available, but again these implant has issues because of inflammation, have some another side effect. So we need a nanotechnology, nano formulation. We did these assay in our lab in Australia. We tested these one. So if you summarize over here, when we use a nano formulated form of these drugs, smaller in size, you can see 56 reach to the cornea, lens 31, and 7% of 30% go to the retina. So you can imagine if you have a nanoparticle to this size, very small in size, which we call as a ultra small nanoparticle, then we can use these nanoparticle ultra small for delivery for different diseases in eye as well. So we developing a lot these molecule in our labs. We tested these one in microfluidic devices, and we developed these tests and tested these one for circulating tumor cell in collaboration with the Chennai here. And we published in a very nice JIP. And here we use the lock nucleic acid septamer put on a chip. And these chip are responsible for taking these stem cell. These are the cancer resistant stem cell from the patient, which we captured from the blood sample. And you can see over when we use these, reuse these for 10 times, still the sensitivity specificity of these assays are still very high. And you can use as a monitoring. So when the surgery is done, after surgery, you can check the number of the cell in these in microfluidic device. You see these devices, microfluidic devices have the these stem cell, circulating stem cell. This is the chip which we developed. And these are the channels here. And this is the sensitivity or the specificity where we use it pre-cancer and the cancer. You can see most of the cancer cell, they get captured with the microfluidic device in our assays here. If you see the patient and the sensitivity specificity, quite high. To summarize my talk or the future perspective for this nanotechnology and nanocapsule is that if you see over a nanocapsule, this nanocapsule is a capsule where we use a small camera on top. We can use compartmentation with this the different drugs. So, for example, somebody have a multiple infection, multiple inflammation, have a cancer as well on the same patient. So you use these devices or these nanocapsule, and you can deliver these drugs because you can monitor, doctor outside can monitor this brand. They can be used as a telemedicine. So where real-time imaging and then releasing these from these nanocapsules at different site, and you clear the infection or these tumor in mice. So these are the study which we are continuing in our hospital here as well. So I think I took a long time, and this is the acknowledgement of the people funding from NHMRC in Australia, from Auckland Cancer Society, HRC from Auckland University, Biodeacon Indo-Australian funding, and from the Deakin University. And these are the centers which are responsible for collaboration. So their list is very long. If you see over here, the list is maybe triple to them. So some of the investigators which I want to acknowledge is majorly from India, Australia, and Auckland. I think with this, I summarize my work, part of the work. And I'm very much thankful to the student or the research fellow who worked with me from the last, especially with the 26 years. Thank you very much.
Thank you, Dr. Kawar, for excellent talk on nanomedicine. We discussed
Thank
the
you.
function of three molecules, lactoferrin, survivin, and VEGF. We also
Thank you so much.
You also discussed some recent economic aspects of nanoparticle in medicine. You also mentioned about the significance of RNA and DNA-based aptamers compared to the antibodies.
Yeah.
Antibodies has problem with autoimmunity and
Yeah,
can
lot of
aptamers.
issues.
Yeah.
Yeah, true.
Yes. We also cited about some oral administration of drug with nano carrier and delivery system for cancer stem cell.
Yeah, we use not only one cancer, we used almost all cancers. Even we continue this work here as well for the urinary bladder as well, and the prostate cancer as well. So we're putting some ICMR grant in future. So I just this May I join here. So maybe next year we hear some more grants or some more funding from ICMR where we can jointly work with you guys, where you can contribute with the nutraceutical. As a nutraceutical or source of natural component for these patients.
Yes, sure, sir.
Thank you, Dr.
Also, one more important aspect you have touched the nanotoxicity in vitro and in vitro methods you have discussed. And above all, from the our interest point of view, you have touched the herbal drugs in the form of nanomedicine. That was very interesting for us. And you have discussed some azadirachta based biopesticide which replace synthetic toxicants. It was a very wonderful talk. And I am sure.
Thank you so much.
I am sure our audience, our students, teachers, and scientists have taken a very good points for their future career or future research. I will,
Dr. Jambulkar, can I add upon?
Yes, sir. Yes, sir. Please.
And I think, Dr. Kambar, your talk is very excellent. And really, whatever we are thinking to have a interface between agricultural sciences and plant sciences, I think you have touched upon the material.
Thank you, sir.
You can have a lot of materials from agriculture, which could be utilized for making these nano carriers. And subsequently, we can look upon not only from the point of nanomedicine, but also from the point of view of development of several modalities for agriculture purposes as well.
Yeah, true. Because yesterday you also mentioned there are some plants which contain the iron more content, so we can use that as a supplementary, because it's not how much iron you are taking, because most of the ladies these days when using oxalate, iron oxalate or iron sulfate, they have a toxicity issue as well, vomit, diarrhea, so many side effects. And natural component, if you're taking as a natural or a herbal product, they are safer and they can absorb through the system very easily as compared to the chemo or supplements.
I think you have very good model system, very good biomarker, and many things where I think we stuck upon, and the lab scale things has to be transferred to the field. Only we are properly testing into the human model. Particularly, you are having a lot of model of this stem cell, or also like HepG2, like the iron toxicity. We have a hepatocyte model. So HepG2 models and other models, they must be available. So at least we can have some of the nutraceutical that can cure the anemia. We have good interest, and also the problems related to bones, the bone disorder. Because if we are able to solve these things, which are really emerging, and henceforth, I think we have to require a kind of discussion so we can.
Most welcome. I want to add over here, I didn't produce the all. We also have a human, we also have a model, animal model for anemia. We also have a model for hepatitis, where you chemically induce and then the liver get this depleted the iron as well. So we have a lot of model which I didn't cover fully all model role. We tested these iron for cachexia. You know, the cachexia is a stage where a cancer patient or the patient who have muscle dystrophy, muscle not function well. So we tested, we have a model, we did all these assay. Yeah, true. We have a lot of model which we can test your product for future.
Okay, sir. I think I am really glad to hear, and I'm sure that we'll discuss, and we can have to have some funding for subsequent work in these directions.
Yeah, true, sir.
Questions
Now-
from the audience?
Now we will have few questions from our participants, students and teachers and parents.
Sure.
Anybody? Anybody wants to ask question, please raise their hand.
Also write in the chat box also. And right now, I think participant, you can ask the questions. That will be good.
Or anybody can ask a question later on. Means if you want, talk to Dr. Anil, and I'm happy to answer any question.
I think there are not much questions, but surely they will discuss with you.
So there are only two things. There are only two things. They didn't understand, or they understand everything.
So I think it is crystal clear, and you have done very nicely all the system which you have developed, and probably the student may be in touch with you because you have already shared your mobile number and the email ID. Thank you.
Yeah, okay, sir.
Yes. Now, to keep the memories of this webinar alive forever in your mind, sir.
Yeah, sure.
Please accept a token of love and gratitude from our side. So I request Dr. Anil Kumar, Director Education, sir, to felicitate our eminent speaker with a token of love.
Thank you, sir.
We feel honored by your gracious presence in this national webinar. Thank you, Dr.
Yep. Thank you, sir.
Thank you very much, sir.
Thank you, Ji. Thank you. Bye-bye.
Bye-bye.
Okay, so it was a nice presentation. So now I would like to invite our next speaker. So now I request Dr. Saharan for his lecture. Before that, I take the opportunity to introduce Dr. Saharan. So Dr. Saharan is a scientist at Maharana Pratap University of Agriculture and Technology in Department of Molecular Biology and Biotechnology, and he is in charge of nanocell facility there. He earned his PhD from Ben Gurion University of Israel. Then he moved to ARO, that is a very prestigious institute in Israel, for his postdoc. He is pioneer in developing phytosyn-based nanomaterials for application in plants having patents of various methods. Currently, he is handling 3 DBT projects on nanotechnology in collaboration with ICAR Institute and other universities. His lab also has a DST-funded international project in collaboration with Iran University. He has published high-impact publications in the field of agriculture, nanotechnology, and guided 8 PhD and 20 master students. So now I request Dr. Saharan to proceed with his. Welcome, sir.
Good afternoon, everybody. I think my voice is coming to you.
You are audible. Dr. Saharan. Please welcome.
So thank you very much, Shant and colleagues and Dr. Anil Kumarji to inviting me for this lecture. And we are waiting for this webinar since long, and now we are on the podium to deliver our thoughts. And with the permission from Dr. Anil Kumarji and Prashantji, I would like to start my presentation.
Please.
Yeah. So thanks for my introductions. My journey started when I was doing a PhD in Israel. And that time we were making some nanoscale material for the Aedes aegypti larvae that is causing the dengue fever. And from that point of view, I learned a lot about the nanoscale materials. And most important thing is that whatever the material that we have designed at that time, in 2003, long back, that was all from the natural sources. So we used the biomaterials for the synthesis of those technology to control the Aedes aegypti larvae, because these larvae, these mosquitoes actually originates in the fresh water. So we can't allow to add any chemicals, toxic chemical to the water. So we developed that economical, the biodegradable formulation to kill the Aedes aegypti larvae in the water. Now, after that, I posted to India in 2007. I joined here as an assistant professor, and onwards, since 2010, I dig into that the nanotechnology field, because that is my background. And since then, it's almost 10 years working on this aspect. And we are working on basically biomaterials, and we want to make some tools to craft these biomaterial into the very valuable materials to be used in agriculture. So everybody is knowing the challenges in agriculture, the global challenges in agriculture. The big issue is the global warming and the population. Now, all together actually make a great concern about the food security. So we need food in coming years, which is what we are producing is not enough. I think we should double our productivity from present point of view, but we have issues like global warming. Global warming is going to play an important role in reducing the productivity in agriculture, and the big share of the global warming is actually introduced by the application of huge amount of agrochemicals in agriculture. More than 5 billion metric tons of agrochemicals are being used per year in globe. And of course, that sustain our productivity agricultures, but at the same time, it also added to the global warming. So at present, we are looking for solutions to reduce these application of agrochemicals so we can protect our air, water, soil. And also at the sideline, the huge application of agrochemicals actually also speed up the resistant development in pathogens. So pathogens start to change their host, change their physiology, and they are going to come in a way that we have to develop some tools to cope with them. So altogether, the message is that how to reduce the application of synthetic agrochemicals in agriculture, and the same time to sustain the productivity. So with this mindset, we have some philosophy, we have some notations, how to deal with that with the small tools of nanotechnology. So every research has some philosophy, some notations to work on. I have 3 points which I am carrying out since long. The first is bio-inspired approaches. So it will bring close to the nature. Second is the things must be multi-mode, multi-targeted. And third is smartly act. So first thing is the bio-inspired approaches. Now, bio-inspired approaches means you have to identify some valuable biogenic material from the nature, which could be used or crafted into the valuable material for our applications. And of course, these biogenic material, which is coming from living organisms, has a biodegradability, and that is good for us, for our environment. The second important thing is that the multi-mode, multi-targeted action. Now, these biogenic material, which is present in plants, animals, microorganisms, they are evolved since thousands of years, and they are screened by the nature, and they are stable in the nature, and they are doing their job, and their function is not a one single. They are acting as a multi-mode or multi-targeted. Fine. So they can go for long-lasting effect and extend the resistant development. Fine. And other thing is growth and protection aspect. Now, all the biogenic materials like Scangy metabolites produced by plants, they have a function in plants, and they are never synthesized 24/7. They are inducible in nature. So it means they are smartly act. Fine. If the plant is facing some disease attack or facing some environmental changes, they start to synthesize some molecules, which is biomaterials. It means they are acting based on a command, based on the demand. Therefore, our notation is that let's find out some biomaterial from the nature and craft them, design them in the way that they can smartly act, so we can reduce the application of agrochemicals, and they could be a very good replacement of the agrochemicals in coming years. And that could be a next generation technology for us. So further we can say, can we mimic the nature to get the solution? Yes, we can mimic the nature, and that could be a sustainable solution. Now, point of view, the plant protections, because I have worked on this plant production area. So elicitors are some of the natural material which is present in plants, microorganisms, etc., like oligosaccharides, protein, peptides, some of the product of AVR genes. They are elicitors, and these elicitors actually excite the plant. They give a signal to plant that, okay, there are biotic abiotic surrounding, so prepare your innate immunity to protect them. Now, elicitors are good material to explore further to increase the plant immunity. The other thing is the plant defense enzyme. There are thousands of defense enzymes present in our nature. They act on pathogens, they act on those fungi, bacteria, which is actually going to attack on your plant. Other thing is in plant, that is antioxidant system. So these are interconnected. Elicitors also interconnected to the plant defense enzymes, and the plant antioxidant system is also related to these elicitors. So therefore, if you are going to search more and more about these biomaterials, which could be a elicitor, which could be a plant antioxidant stimulator, which could be a systemic acquired resistant stimulating molecules, we can generate a new tools. So overall, that maintain the cellular homeostasis and the plant become healthy and protective from the environment. So what I'm using in my lab since last 10 years for my research, so all are in biogenic materials, like chitosan, the foremost backbone in my lab, which is used for making so many nanomaterials that is biodegradable, biocompatible, and eco-friendly. Likewise, we have a copper, zinc, silica, Fe. These are the micronutrients, which is act as a cofactor for plant enzymes. We are also using these to make some composites. We are also exploring the plant essential oil components, that EOCS, that has a great bioactivity against the pathogens, against the microbes, and act as a defense molecule for the plant. There are some plant glycosides, which is very promising for defense mechanisms. And likewise, some natural acids like salicylic acids, which is present in the plant, and it is a good signal molecule that communicate between the cells about incoming danger of the pathogenic attack. So these are molecules actually we are exploring in our lab for making various nano formulation. Now, first of all, I'm going to touch about the most important biopolymer that we are exploring, that is the chitosan. Now, chitosan is very similar to chitin and cellulose. The difference is only one functional group. In chitin, you have acetyl group, whereas in chitosan, we have a NH2 group, and whereas in cellulose, we have OH group. Now, this NH2 group, which is present in chitosan, actually provides adding to its bioactivity. Now, these material like chitosan or chitin, we can extract from crustacean, exoskeleton, fungi, bacteria, mushroom, and they are biodegradable. Biodegradable means when this chitosan is going into the nature in the soil or in the biological system, the enzymes present in the living organism, they digest it. They act on the glycosidic bond of its units and separate into their components. So this is very valuable for us. So it becomes more compatible to us and non-toxic for the environment. Other important aspect of the chitosan is that it acts as a very strong plant elicitors, induce plant growth and defense. And it is considered as a plant growth regulator. At the same time, it has a great antimicrobial activity against the fungus, bacteria. And the problem is it is water insoluble. It is one of the negative points that chitosan is containing. But at the same time, it is cationic in nature. It has a positive surface charge. So it has a great affinity towards the biological membranes to attach on. And other thing is that it is acid soluble. So if you want to mix the chitosan in the water, then you have to make the solution acidic, around 2 to 3 pH of solution should be there, then chitosan can be mixed easily there. So this is a drawback of the chitosan. Now, with one or two drawback, chitosan has been modified chemically by many groups. You can find out in the literature that thousands of modification has been done in the chitosan by replacing its functional group. So you can replace the NH2 group, you can add a methyl group, you can add alcohol group, you can add ammonium ion, you can add triple R with a different methyl sulfur groups. So these are substitute chitosan, and people are claiming that the chemical modified chitosan can easily be soluble in water, and that is also true. There are chitosan available in the market which is chemically modified; they are soluble in water. But the big issue is that when you are going to modify the structure of a chitosan, native structure of chitosan, by replacing the functional group, its biodegradability may be reduced. That is the main issue of a concern, because why we are taking chitosan? Because chitosan is biodegradable. We can use it as a carrier, can a delivery system. But once it loses the biodegradability, then there is an issue. Fine. So to meet these challenges, we have worked on to design the chitosan into the nanoscale so it can easily mix in water. It gives a very good monodispersed colloidal system, so that can be applicable to the agriculture. So normally chitosan is a linear polymer which has a 50 to 190 kilodalton of a size. So it's a big size biopolymer. And through nanotechnology, by bottom-up or top-down approach, you can convert into the symmetric structures that may be compact, stable, and also nanoscale size. And it gives a homogeneous colloidal in the water. And so it is easy to apply to the agricultures. And by this bottom-up, top-down approach, you can customize the shape, size of the chitosan linear biopolymer into the spherical structures or rod structures or star-shaped structures. So by using the nanotechnology tool, you can cope the problem of the solubility of the chitosan. Even the big linear biopolymer of 50 to 190 kilodalton can be reduced to a nanoscale size, and that can be applicable for our purposes in agriculture. Now, by this bottom-up of top-down approach of the nanotechnology, the shape, size, the surface, the morphology, the physicochemical properties can be play around. Like you can increase the surface charge on the chitosan, you can decrease the surface charge of chitosan, you can decrease the pore on the surface, the pore volume of the surface according to your requirement. So altogether, when you are going to design the chitosan, nanochitosan structures, they are actually a accumulation of 0D nanoparticles, which has all the dimension in nanoscale, but overall they are actually 3D material. When we are going to classify the nanomaterial based on their scale. So with this thing, you can also blend these structures with any active ingredient, like you can use micronutrient, you can use some acids, you can use some drug also. In medical field, I think chitosan has been explored a lot for drug delivery vehicle. So you can encapsulate drug into these case-like structures, and you can change the pore size, you can control the release profile of the encapsulated material into these in these nanochitosan structures. Now, this is one of the example that how chitosan can be used for agriculture. This is the very simple presentations which we have. This work has been done in 2015 by us, by one of my student. So at this side, you can see a linear chitosan biopolymer, and we convert through the nanotechnological tool into the compact structure with a nanoscale. And further on, we encapsulated copper inside these structures to make a composite structure of the chitosan, which carry copper ions. Now here, the thing is, these structures are in nanoscale, but the copper is not in nanoscale, that is still in the native ionic stage, which we want. So therefore, in these structures, copper is not in nanoscale, and we call it copper chitosan nanoparticles. So here the chitosan structure actually is a nanostructures. Ions are very smaller than the nano and then encapsulated inside. Now, by changing the various parameters, you can play around. You can make more compact structures. You can make more small structures. You can increase the encapsulation of the copper by changing the parameters. That depends on you, what you are looking for, how much copper you want to encapsulate and how much copper you want to deliver to the plant system. So likewise, you can encapsulate so many micronutrients into these structures. Not only micronutrient, you can encapsulate other pesticide or fungicides, other acids, like we have also encapsulated salicylic acid to generate the plant immunity in the plant by spraying these nanomaterials. So this is a simple presentation how you can design the linear biopolymer of chitosan into the nanoscale nanoparticles. Now, if we are going to encapsulate these micronutrients like copper, zinc, silica, Fe, salicylic acid, what actually is going on? Now, first of all, now the purpose is of slow release or sustained release. So plant can get these materials for a long time and with the minimum doses. So they can use it properly and there may not be any antagonistic effect or response from the plant, and they get a sustained supply with a low dose, low cost, and also the chitosan backbone in these structures act as elicitors. So this elicitor also provide a protection from the microbes to the plant. So therefore, this whole structure is acting in a way of multimode. It has also a elicitor activity. It has some micronutrient for supplying of nutrient to plants. So overall, it acts a full package to the plant for maintaining a very vigorous cellular homeostasis. This is the purpose of the encapsulation of any active ingredients like micronutrient or other thing for plant. Now, the application of these nanomaterial, which is encapsulated with the active ingredient, is by seed treatment and by foliar application. Now, seed treatment is important, that what we have doing since last 10 years, that when you're going to treat your seeds, they make a coat, some of the particles goes inside the seeds, and that protect it from the seed and soil-borne diseases. They are not allow any fungus to go inside during the germination period. And also they act on the reserved food which is present in the seed, remobilize them and increase your seedling growth. So that increase the seedling vigor index. This is the purpose of seed treatment. Now the foliar application further, the purpose is to provide the nutrient at a key stages of the development. And also because chitosan is there as a backbone, it provides immune elicitors for the plant, and plant can start strengthening their innate immunity through synthesis of antioxidant enzymes, defense enzymes, fine, to make a shield for incoming diseases. So therefore, it is just like act as a vaccine for the plant for incoming disease, which may come after 1 week or 2 weeks when we're reaching to the flowering stage or seed filling stage, likewise that. So therefore, the treatment of these nanoparticles is through seed treatment and foliar applications in the crop. We have tested in the maize crop, we have tested in tomato in 2015-16, and we are also applying on the wheat crop also for the action of these materials. Now, these materials have been synthesized in our lab, and we used to characterize by different methods, because in agriculture, most of the chemicals we used to mix in water. And these materials are not, we are not applying in the soil, because when we are going to throw these materials in the soil, they are biodegradable in nature, so they won't act. So the application recommendation is only through seed treatment and foliar application, so that each and every portion should reach to the plant cells. Now, characterization is important. So we used to characterize everything, also in dry form, also in the liquid form, because after all, the material will go in water and applied in water mix to the plant. So how it react when it is in dissolved form in a water? So we used to do dynamic light scattering. There in that we can check the monodispersity. We can check how many particles present in the water in which scale, how many percent of particles having a size of 200 nanometer or 300 nanometer. Either they are monodispersed or polydispersed. What is the surface charge of particles? Surface charge is very important for agriculture. If your particle is positive surface charge, high positive surface charge, they can attach strongly to the plant surfaces, and we can cope with the drip off effect. Otherwise, most of the agrochemicals we are supplying on the crop, the 20 or 30% they're going into the soil by drip off effect. So therefore, you have to maintain the surface charge. So here, DLS technique is used to check the surface charge. Further, you can check the internal structures, the external structures, the elemental compositions through EDX, XPS you can do, crystal structure you can do by XRE techniques, and the encapsulation. That is very important. If somebody is claiming that I have a formulation which do a slow release of a zinc, so it has to be proved that how much zinc actually encapsulated into the nanostructure. So 1 gram of nanoparticle, how much zinc they are containing. So that should be classified. That is done by atomic absorption spectroscopy for characterization of the material to check the encapsulation efficiency of the particles with these micronutrients. So you can see some of the picture of the dry powder that we used to prepare in our lab that stay in the dry form, and you can store it in room temperatures, and whenever you want to use, you can mix in water and you can use in the field as a seed treatment and as a foliar agent. Now here, the most important the characters of these materials are that they are smartly act on the diseases. This is the most important characters of these formulations. Like when we are talking about the copper encapsulated chitosan nanoparticles, in which copper is encapsulated, covered by the chitosan nanomaterials, and copper is known for antifungal agent, and chitosan also itself is antifungal agents, and it gives a slow release. Now, this is important when you have a intense disease in the plant. So the cellular environment of the plant where fungus actually intensely infected your plant cells, their structure of the cell is disturbed, and the pH of the whole system goes towards the acidic side, because so many fungus, they are producing acids. Now, as I told you, that chitosan is soluble in acids. When it touches the acids, when it meets to the acid, the chitosan structure dissolves, and immediately your encapsulated material comes out and acts on the fungus. So this is the pH responsive smartly action of these material. Fine. In my second slide, I mentioned that on command, on demand. So this is how it follows. Fine. If there is enough infection on the plant cells and your material is there, so normally it slowly releases the copper in cellular environment. Once it meets to the acidic environment, the release is become faster. And when the copper comes out, it starts to act on the fungus. So this is how a smart act of the chitosan nanomaterial. Now, as I told you that what the seed treatment also increased the seedling vigor index. Now, how it possible? So we dig out this reason also in 2016. We checked and we proved it for coming years. When you are going to treat your seeds of maize, we have checked in maize, also we have checked in wheat. What happened? Actually, the amylase which is present in the seed, protease which is present in the seed, activated by treatment of these materials, and these amylase act on the stored food like starch, and protease act on the protein which is present in the seed. Because for seedling growth, they needs amino acids and energy. Now, these amino acids comes from the dissolving of the protein through protease activity, and the energy will come from starch. So these amino acids and free sugars, they are now available more compared to the normal seedlings and increase the seed vigor index. Fine, seedlings are very healthy, and the seeds are germinating very fast when they are treated with these nanomaterials. So we find out that on the treatment of these nanoparticles on these seeds actually increase the stored food remobilizations. So stored food is used. Otherwise, what happened when you are going to grow the maize seeds in the field, you will find after seedling growth, still some starch is remaining in the seed. But when you are going to apply these particles, I think nothing is left. Every portion of the reserved food is used in seedling growth, and you will have a bigger seedlings when you are treated these particles to your seeds. So this is actually the biochemistry of a seed treatment. Now, as I was talking that these nanostructures of chitosan can be encapsulated by several active ingredients, and one of them we tried the salicylic acid. Now, salicylic acid play an important role for plant production point of view. When the plant is showing the resistance against any pathogens, check the amount of salicylic acid. Salicylic acid is present in the plant in a good amount at that moment, because these molecules actually act as a signal molecule from one cell to other cells. Fine, and it is also involved in so many pathways which provides strength, increase the metabolism when the plant is under disease conditions. Fine, but if the salicylic acid is not produced properly at a proper time, now the disease infection is becoming more and more. Fine, so salicylic acid is one of the very important compact, not only with the disease production but also for the abiotic test. Fine, so put into these in the mind, we have designed one material in which we have encapsulated these salicylic acid into the chitosan nanostructures to make a slow release system. Now, if you go to the literature, if you go to some recommendation of some institute, they have recommended that this much amount of salicylic acid you can apply as a foliar, and you can see the good results. But the dose is not fixed. Why it is not fixed? Because the salicylic acid, endogenous salicylic acid amount is always fluctuating. If you're going to supply more amount and plant is already having salicylic acid, you will have an antagonistic effect. You will have some response which is going to backfire you. Fine. Therefore, you must have a slow release system by which you can sustainably supply the salicylic acid to the plant, and that cannot cross the limits of the acceptance of the plant. Therefore, we encapsulated this salicylic acid into the chitosans and make it a slow release. And we found a very wonderful results, and we have checked this formulation in the field against the PFSR disease of the maize. And we all classified all the cellular response, physiological responses, the biochemical responses. And one important feature that we found that the chitosan and salicylic combinations actually also make the cell wall of these the plants very strengthful, reinforce the cell wall. And what we found that the reinforcement of cell wall is because of the lignin deposition. Now, lignin is very important. If your plant has a good amount of lignin in their vascular system or in their stem, I think they are resistant towards the fungus. They are resistant to the bacteria. Because in PFSR disease, we found that the fungus actually blocks the vascular system of the plant, and plant is not getting the food to a proportions, and plant start to die. Fine. So if your lignin deposition is there in the cell wall of your vascular bundles and area, I think fungus is not able to penetrate that. And this is one of the reasons that these formulations act very nicely against this disease in maize. And this is what we carried out for next year also, and we found the similar results. So 2-year field trial we checked, and this field trial was contained field trial. That is not an open field trial, because all the nanomaterials which is we are applying outside, it should be contained manner, because still the nanotoxicology study or the recommendation from the government of India is still saying that until you are not going for any nanotoxicology study, you have to make all the contained field trial. So these are some of the results that we have checked. You can see the pictures, the lignin deposition here, and you can see the control of disease by application of these salicylic acid encapsulated chitosan nanomaterial. Other thing I was talking about that how the immune system is strengthened by application of these nanomaterial. Now these nanomaterial act on most of the antioxidant enzymes like SOD, like POD, like PAL, CAT. Now when the plant is under disease condition or any abiotic stress, start to produce reactive oxygen species, and this reactive oxygen species start to interact with your enzymatic system and your plant metabolism goes down. Therefore, you must have some strategy to uphold these antioxidant enzyme activity, so they can swap out all the reactive oxygen species which is built in the plant during the infections. And what we found that application of these chitosan-based nanoparticle overall, they increase the enzyme activity of these antioxidant enzymes by 2-fold, 3-fold. And that is the main reason how you can protect your plant from the disease. Not only they are acting these particles on directly to the fungus, but also they strengthen the plant innate immunity by upholding the antioxidant enzymes. Likewise, we have checked the content of reactive oxygen species after application of these nanoparticles. We found that it has nicely balanced the amount of reactive oxygen species. Like the most dangerous reactive oxygen species in the plant is a hydroxyl ion and superoxide. They are most dangerous. So these enzymes, what they do, they convert these reactive oxygen species like superoxide into H2O2. That is hydrogen peroxide, which is a signal molecule and which is passed from one cell to other cells to tell that, okay, the infection is started. Now you have to build up your immune system. Fine. So these nanoparticle application actually balance these reactive oxygen species in the way that they are not dangerous for the plant, and plant can withstand the disease when there is an immense attack of the fungal or bacterial disease. Now, in same platform, same template, we also developed that zinc encapsulated chitosan nanoparticle to check its effects on the crop. And we checked this is in the Curvularia lenta, that is giving a CLS disease in maize, the fungal disease. This is the type of a foliar disease. And we applied this zinc encapsulated chitosan nanomaterial through foliar and also for seed treatment. And we found very good results. We checked the leaf morphology and the anatomy, and we found that it also increased the lignin content in the vascular bundle. You can see the pictures, and that actually protect the leaf from the infection of the fungi. Not only that, it also increased the amount of antioxidant defense enzymes like SOD, POD, PAL, PPO, lignin content, and it also acts to remobilize food from the leaf area to stem towards the grain filling. Fine. This is the new thing that we got from this research, and we published this research also in a good journal. And this is a stepping ahead that these nanoparticles not only protect the plant from the disease, but also help in remobilization of the food from the leaf to the grain filling. Fine. So leaf act as a source organ. And if the proper the translocations happening of the food from the leaf to the upper portion, I think your yield may also increase. So these are the footsteps that we continuously, this work we have published in 2019. We started this work in 2018. We did the 2 years trials of these materials, and now we are trying these material also in the wheat crop for the source activity in zinc stent. So this is a slide I was talking about, that on command, on demand. Fine. When these particles are applied on the plant and plant has already infected with the fungus, they act more strengthfully because the chitosan is dissolved in acidic medium. Fine. So you can see the structures here. at the pH 4.4 or pH above 4.4, the structure is dissolved, your copper ions comes out and act on, and we checked it through the in vitro assay that how long it will take of slow release, how long it will take to rupture all the chitosan nanostructures. So that can be compatible for the application in the plant system. Now, when we checked all these material in the field, what we found that, as I mentioned, that not only it protects plant from the disease, but it also acts as a source of nutrient to the plant. Right? So this we have reported in our paper in 2020, that the structure of a chitosan, which is encapsulated with the active ingredient, can supply nitrogen, phosphorus, and the active ingredient like copper or zinc. It means it is a full package. It means it is a composite which can act as a functional fertilizer because it provides the macronutrients and it also protects plant from the disease. Therefore, this is term that we have coined, that is a functionalized fertilizer. And these are the things is in the future. In coming futures, we will have these type of formulations which not only act as a fertilizer, but they act as a biostimulant. They can protect your plant from the disease and they can work dynamically. Fine. So there is no need to separately put this one, this one, this one. One composite structures can provide in future overall protection, growth through supplying the nutrients to the plant. So this we published paper that is the chitosan nano fertilizer that foster the source activity. Fine. Source activity means it increase the photosynthesis of the leaf. It increase the food remobilization from the leaf. One thing is important, when plant under stress, what happens? Photosynthesis is there. You have enough photosynthesis, but the food which is reserved in the leaf are not translocated towards the grain filling and they stop in between. So under stress, if you're going to apply the material, we found that this help in remobilization of the food towards the seed, which is in a filling stage, and you will get a higher yield of that. Now you can see the whole how it actually function. These functionalized nano fertilizer, which is actually made up of copper, salicylic acid, and chitosan. Through the foliar seed treatment, you can see source activity, which is actually regulated by the antioxidant strength of the plant, regulated by the chlorophyll content, regulated by the photosynthesis rate, regulated by the food remobilization. So how much food actually remobilized from your leaf toward the stem to your grain. If the food amount is remobilizing very nicely, then there will be a very good balance between the source activity and sink strength. If your source activity goes down, there will be no any sink strength. Fine. So they are going to deplete with the food. And when you have a higher source activity, more food is produced, more food is remobilized towards the seeds. Your seed weight is high, your starch content is high. This is how we have give a hypothesis on that, and we have proved this one through application of these functionalized nano fertilizer, which is made up of chitosan, copper, salicylic acid, a combination. Likewise, we have also developed one silica encapsulated chitosan, which is highly porous structure we ever seen in our research. It has the pore more than that what we have found in the copper and zinc based chitosan nanoparticles, and we applied in a maize just to check what is the effect on the yield and other thing, and they are very promising. The important thing is that source activity is really induced by these material more than that of zinc and the other formulation that we have tried. So, and we have given so many hypotheses in that paper that what are the reasons why these silica encapsulated chitosan is doing this job, right? So this is also a very promising material that we have developed, and we used to say it chitosan silica nanofertilizers that enhances the plant growth and yield in maize. And here also we use the seed treatment and foliar applications. Now, based on all these years' work, one project recently sponsored by DBT to us, due to the corona, it is actually delayed. But we propose them to do a multi-location trial of these functionalized fertilizer of chitosan based, which provides not only micronutrient but also macronutrient like nitrogen, phosphate. And we are happy that this year they are going to give us some money, and we are going to apply. We can do a multi-locational trial in the contained field in Haryana Agriculture University, also Bangalore, in our center also. And these are the material that could be a next generation formulations and platform for the newcomers. Those can be explored, these biomaterial as a fertilizer in the crop. So we can decrease the application of the traditional fertilizer, which we are using a lot. So we can protect our environment by introducing these biodegradable material to the field. And most of these material is applied as a foliar, not in the soil. Now, one point that I mentioned in my third slide, that we are also exploring the essential oil components. These are immense, they have immense bioactivity. We have thousands of essential oil components present in oil. And one of the components that we have identified that is present in thyme plant, ajwain ke plant mein hota hai, that is thymol. The big issue with the thymol is that it is not also mixed properly in water. If it is mixed, then it is not stable. It may be making a cream, or it may make a big droplets. And when you're going to apply to the field, I think they are not homogeneously applied. Fine. So people used to mix it with the surfactant, high amount of surfactant. And surfactant is not good for our health also, or also the plant health. They are just to mix in the water. Fine. Therefore, what we have done in this, and this technology is also patented, Indian patent, we got this of this technology. So we use a surfactant, very less concentration, which is also from the plant origins. So what we prepared nano droplets of this thymol and mix in the water and apply to the plants to get resistant, to act as a fungicidal, bactericidal activity. And it is also, we found that there are some signals that insect is also coming less when they are these formulation applied on the plant. So based on these findings, we have submitted one project to the DBT that is in before COVID-19, and I think this year we are going to get a grant of that we want to explore these formulations to fall armyworm of the maize. That is the biggest problem at present in India in the maize. So this is a very promising formulation that we have developed, and we have make so many changes in the formulations. And we have also made this formulation which also carry the micronutrient to the plants. Like we coated these nano droplets with the zinc, we coated this nano droplets with the copper, magnesium to apply to the plant, that not only protect it from the disease, but also at the same time apply the micronutrient to the plant. All this has been published after getting a patent. We have published, I think, 2 or 3 papers on this aspect of these thymol nanoemulsions. Currently, we have 3 patents in our name based, that is the thymol and some of the chitosan. And currently my lab is funded by DST DBT funds. We have 2 projects recently given by the DBT. One is the upscaling of chitosan and thymol based smart nano formulation to control the fungal disease and fall armyworm in a maize crop, that is sponsored by DBT. And we have a collaboration with the Indian Institute of Maize Research Ludhiana, CFET Ludhiana, and TNU Coimbatore. So these are our collaborators. And other project that we got, that is on the nano fertilizer. That is the upscaling and on-farm evaluation of the biodegradable nano formulations, that is sponsored by DBT. And in this project, we have a collaboration with the HIU ICAR and the NABIR Bengaluru. These are some of our publications done by our students and PDF fellows from last 5 years. And on the behalf of, I acknowledge to my team And because of that only it's happening, my past students, my present student and incoming students. And I also acknowledge here your university to invite me to share my views. And so the students, scholars, they can learn things, they can develop their own idea, and they can fulfill the things, and they can give some inputs to the Indian agricultures. Thank you very much.
Thank you, Dr. Saharan. It was an excellent talk on nanomaterial for plant protection and growth promotion. We discussed bio-inspired approaches to develop the biogenic nanoparticles. We also mentioned about the chitin, copper, zinc, silica, ferrous, essential oils, and plant glyphosate having different mode of action. We also discussed about in detail chitin as a biogenic material having different mode of action. In your discussion, you told about the use of chitin as biogenic material. How to get rid of that drawback was wonderful to know that it is insoluble in. So to prevent it, we have to solubilize it in acidic condition. You also discussed about the customized nanoparticle and their use as a seed treatment and foliar application against the various crop diseases. That was wonderful. The role of salicylic acid, functionalized chitin nanoparticle for disease management, and its so slow and sustained release. That is the need of the hour of the salicylic acid. As we all know, you have also discussed in your talk that we at random we cannot spray salicylic acid because plant has its own demand. So this work, like its slow and sustained release of the salicylic acid, will be helpful to the plant to fight it against the various pathogens. Another finding is the slow release of zinc chitin against the Carvolaria lunata as a foliar spray. That is also a path-breaking findings of your experiment. You also you also mentioned that the application of your copper chitin nanoparticle plant remobilize the food in plants. And you have mentioned about the biostimulant plus fertilizer, which together may act as a functionalized fertilizer. So I congratulate you on another patent of USC nano emulsion of thymol, which you have told that you can use it as fungicide and insecticide both. But this is another feather on your cap. See, it is a need of the hour, as you have mentioned that you will be testing it against FAW. So I wish you best wishes from all of us that you will get success in this area. Now the floor is open. There are many questions I feel in the mind of the audience. So questions are open to Dr. Saharan. Audience, please. In the chat box, first we can see there are many questions. First is from Revti. Sir, any nanoparticle to reduce viviparity in soybean crop due to the effect of post-rainy season? Cost of that recommend nanoparticle where I can get?
Okay, so I don't have much experience related to what the student has mentioned. It should be tested. If some material is there, like ours, if we have any invitation, something like that, any university that okay, we want to test it and other things, and we can provide. They can test it. How actually it going to interact with those. Like if it is a type of an insect, then chitin will not work with that one. Then you have to use the other formulations like what we are going to trial in coming 2 years, the fall armyworm against the thymol emulsions, because we found that yes, it has some activity, but in chitin, I don't think so that it may work against the insect because chitin is totally different nature. So and other thing is that I don't know what other groups are doing. Maybe they have some formulations to cope with the problem that what student has mentioned. But we never checked this one. But nanoparticle, of course, can be tried. In coming years, the only option is now in nanoscale material. So this could be done.
is from Dr. Sevan Nayak from ICAR. He wants to know the name of nanoparticle or nanomaterial for enhancement of photosynthesis. Don't know.
Okay.
Dr. Saran, you are please unmute. Please.
Yeah. What I understand from the question is that they want to know how nanoparticles actually the mechanism to increase the photosynthesis, right?
Yeah, that is a question, but he was asking about the nanoparticle or nanomaterial for enhancement. Oh, it's one and the same. Yeah, please.
Okay. So in my view, what we have done, we used to check the chlorophyll content first of all. If we are going to apply any material to the leaf or the plant, we check chlorophyll content. If chlorophyll content is higher, of course the photosynthesis rate will be higher. Fine. Now, why the photosynthesis chlorophyll content is higher when you're applying something? Simple is that if plant gets micronutrient in very balancing amount, sustained amount, that is also going to increase the chlorophyll content. Fine, like you can use zinc, you can use copper, magnesium, fine. Even silica, what we found in silica chitosan, the photosynthesis is much higher than what we achieved in zinc and other things. Now, the reason behind that is not clear biochemically, but we checked one paper published in Nature way back in 10 years. They used silica, normal SiO2 on the plant, on the maize plant, and by deposition of this silica on the maize surface, maize leaf surface, they help in absorb more light from the sunlight, and that is more light is absorbed by the leaf, they increase the photosynthesis rate. Fine. This could be the reason of what we have tested in our things. And there are some particles, nanoparticles developed by some group in US, and they are providing like magnesium or something. And you know, in a chloroplast, I think if you have an ample amount of these magnesium, zinc, because zinc act as a cofactor for more than 300 enzymes, which is related to the plant metabolism. And directly, indirectly, they are also going to strengthen the chloroplast structures. If chloroplast structure is strengthened, the pathway is more smoothly and you will have a good photosynthesis rate. This is what the explanation.
Thank you, Doctor. There is one question from Bharat Baburao. Yeah, you can ask question.
Sure, sir. Thank you very much, sir, for a nice talk, and it's very much informative. I would like to ask about chitosan nanoparticles. So you are preparing chitosan nanoparticles using the sonochemical method, that is sonicator. So what are the actual changes take place after sonication?
Okay, no.
It may be reduction or it may be fragmentation. Or
Okay, so sonication is a small event in the synthesis process. It is not only ultrasonication. First of all, we are going to cross-link these linear biopolymers through cross-linker, that is TPP, trisodium polyphosphate. Fine. These cross-linkers bend this linear structure of chitosan into spherical shapes. Fine. And once we got these compact structures, which may not be in nanoscale, they may be in macro scale, then this solution actually come under the ultrasonication to disintegrate these agglomerates, fine, just to separate these big particles and bring the system in dispersed form. Fine. So ultrasonication, the function of ultrasonication is to break the agglomeration. Fine. But the heart of the reaction is the cross-link that we use to do. Fine.
okay, thank you. Thank you.
Thank you. Dr. Saran, there is one interesting question from PA. Dr. Rupesh Kumar Arora has asked, is chitosan work against the sudden wind or parawind? Means in abiotic stress in cotton. We know in Baswara also we have seen parawind.
Okay, this is a type of a disease, no?
Yeah, parawind is a physiological disorder.
Okay. So
Abiotic.
no, I am not aware of that. I never, I even don't know ABC of that. but I don't know what is the mechanism. First of all, say for example, in general, I can reply like, first of all, you have to check what actually the physiological event happening of any problem in the plant, whether it is disease or whether it is other factor of environmental, fine. You check the causes, fine. Who causes that, fine. Say for example, if some disease or some like what Dr. Prashant is mentioning is because of some of the deficiency, fine. So we have to target those deficiency, fine. Say for example, it is a type of environmental effect on the plant because of the high temperature, low temperature, freezing, or some deficiency, or some the impurities in water supply or something like that. We have to check those events. Then we can check whether your material which is designed like this going to act or not. Fine. This is just like a question of photosynthesis. Now, if you're going to apply normal zinc sulfate in the plant, your photosynthesis rate will be increased. Now, there is no direct link of zinc to the photosynthesis, but there are so many events which makes the link. Fine. So you have to check first of the physiological events, a biochemical event of that cause, and then we can target those points one by one by any tools or any technology.
Right. Thank you.
I think
Now I request Dr. Anil Kumar sir to interact.
I think very excellent talk, Dr. Saharan. After a long time, you delivered one of the seminar, one of the lecture at Pantnagar. That was
Yes,
another brainstorming session. And indeed, I was really happy to listen once again and your innovative work,
Thank you, sir.
particularly in the technology area. And I think the question asked by Dr. Rupesh Kumar Arora is very well answered by one of the technology, that is a silica-based chitogen. And you know, silica is going to play very, very significant role for protecting against many of the abiotic stresses. And it has been proven that silica is providing protection against many of the, whether it is a heat stress, whether it is a dot, and so on and so forth. So I think it could be tested in this para-built in cotton. So I think these particles have a lot of potential. Only thing is systematic study has to be required. And my question is, you know, this chitogen, the backbone structure of the chitogen, that is a lot of variation. And you know that once it is hydrolyzed to give chitin fragments, then it is inducing the defense signaling pathway, and ultimately it is providing the immunity. So my question is, what are the chitogen source you are using? Whether chitogen source are really also vary, and then the properties will vary, and then subsequently its effect, whether it is immune induction or maybe in the yield increase or maybe nutritional quality. This is
Okay, thank you. Thank you, sir. And sir mentioned that I have been in Pantnagar in 2012 or 13, I think, for a lecture. And at that time, Dr. Anil sir is there. And very interestingly, I want to mention here that one of the theses on the thymol nanoemulsion review was Dr. Anil sir, and I still remember that. And now comes to your questions of the chitosan sources. Now you are rightly said that because this is a biopolymer and this is actually produced by chitin, and there is a lot of impurities found in a chitosan powder which we are purchasing, which contains the chitin. So the 80% material in the chitosan is actually a pure chitosan. Rest is actually chitin, because the difference is only the acetyl group. Fine. So 80% it contains the NH2 group and 20% acetyl group. So there is impurities here. To cope with this impurity, we actually use for our synthesis purpose, for small amount, we use sigma chitosan. And they claim that it has molecular weight from 50 to 190 kilodalton. Otherwise, there is a huge variation of molecular weight of chitosan. And you rightly said that the molecular weight of chitosan really affects a lot for your designing these nanoparticles. Fine. If it has a more chitin content inside, then you won't get a compact structure. You won't get a symmetric structure of this one. Therefore, to avoid these things, we use Sigma. It's costly affair. But if you are going for a much larger scale or scaling up these materials, then you have to search the cheaper sources, and they are available in India also. Nowadays, chitosan is produced a lot by so many seafood industries in the seaside, like Kerala and other thing, and also in Gujarat port. Fine. So you perfectly said that the source is very important. Now, the source that Sigma is actually providing, that is the isolated from the crustacean exoskeleton. That is not from fungus. And according to research, that is more cheaper. Isolation of chitosan or converting the things from the fungus is much higher pricey, because whatever you want to develop in agriculture, it should be cheaper, economical. If the backbone is so costly, then I think it's very costly for the farmers, and farmer will not adopt any technology if it is costlier than what they are using nowadays. So the source of a chitosan is very important, and we are very careful. That's why we are only using a Sigma for our research purpose. Fine. So the molecular weight remain in some category, like not so bigger. Fine. If chitin is mixed, then it may create so much hurdle in making the nanoscale structures, the customized structures.
I think excellent talk, and I hope that newly developing university, Dr. Prashant Jambalkar, very hardworking and sincere, and he is trying to take nanotechnology flag as a plant protection system. So I'm sure that he may interact with you, and maybe some of the things he can also start over here. Thank you very much.
Thank you, sir. Thank you for your words. And I'm looking for such type of collaborations from your university, like Jambalkar sir is working there, and we are open for any collaborations. And this is actually, this type of platform is only for these interactions, to just scatter your knowledge to the new students, the scholars, so they can develop their own idea. Thank you very much for your invitation, sir.
I think I'm coming to your university probably in 1920th, and
Okay.
detailed discussion. And
Yeah, sure, sir.
hopefully time is very short. Maybe after only the PhD YOYC examination, I may have some interaction.
Okay, sir.
Visit your lab. Subsequently, my personal visit is to have a family tour to Rajasthan. So we'll meet it.
Okay, sir. Welcome, sir.
Okay, thank you very much, sir.
Sure.
Thank you very much, sir. Now, please accept a token of love from our side. Now, I request Dr. Prashant Jambalkar to felicitate our next speaker.
Tanuj, please.
Dr. Tanuj.
Thanks a lot, sir.
Thank you, Dr. Saram. Yeah, we will be in touch.
Okay.
Thank you. Bye.
Okay, in this series, now I welcome Dr. Ashok Kumar Nadda. Ashok is a good friend of mine. We studied in same university. So good to see you here in the same platform, Ashok. So before his lecture, I would like to introduce Dr. Ashok. Dr. Ashok is working as an assistant professor in the Department of Biotechnology and Bioinformatics at JP University at Solan, Madhya Pradesh. He holds an extensive research and teaching experiment in the field of microbial biotechnology, with research expertise focusing on various issues pertaining to nanobioanalysis, microbial enzyme, biomass, bioenergy, and climate change. He holds international work experience in South Korea, India, Malaysia, and China. He worked as a postdoctoral fellow in the state key laboratories of agriculture microbiology in China. He also worked as a brain pool surgeon, assistant professor at Konkuk University, Seoul, South Korea. Dr. Nadda has published more than 135 scientific contributions in the form of research review, book chapters, and other journals of international review. The research output includes 9 research articles, 30 book chapters, 16 books. He is main series editor of Microbial Biotechnology for Environmental, Energy and Health, that publishing the book under Taylor and Francis from USA. He is also a member of editorial board and reviewer committee of various journals of internal review. Thanks for accepting our invitation. Now I welcome Dr. Nadda for his presentation.
Okay, thank you very much, Dr. Piyush. It's very nice to see you after a long time, maybe more than 15 years, I think.
Yeah.
Is it? So not maybe 12 to 13 years. Okay, so hello everyone, very good afternoon. First of all, I would like to thank all the members of organizing committee and Vice Chancellor of Rani Lakshmi University, Professor Arvind Kumar and Professor Anil Kumar for inviting me here. So before starting this, maybe I can share the screen. I hope that my screen is visible. Slides are okay.
Yes.
Okay, so we are already late. So without wasting time, I would like to just jump to the topic that we are going to discuss. So thank you for inviting me in this national webinar. So my topic is nanocatalysis for enzyme immobilization and environmental remediations. So where we are located and working recently, I am working in this university, JP University, which is located in Himachal Pradesh, just near to Shimla, 20 kilometers away from Shimla, one of the beautiful campus in North India. So I welcome you all people. Sometime if you get the chance, you can visit here. Area of research interest. I am working in multidisciplinary field. So our area is gene cloning, expression, directed evolution, site-directed mutagenesis, 3D structure guided protein engineering, homology modeling. That is the base of our research. Then we are working on the microbial enzymes, and we try to improve the activities and stability of the microbial enzymes by various techniques. So improved thermostability and activity of the enzymes by immobilization techniques, synthesis of nanoparticles for immobilization of enzymes, protein inorganic nanohybrids and nanoflower, nanobolt, nanorattles, so many structures by using the enzymes that we can synthesize. Then application of these biocatalysts for CO2 conversion, carbonate synthesis, biofuel production, biosensor developments, and various products of commercial use. That the area we are working on. Why CO2 conversion is important? I saw that there is some bar on the slide. Can I remove this by some way or maybe hide this? Yes. Why CO2 conversion is important? So uncontrolled emission of CO2 from various sources that impart to global warming. We all know that the absorbed CO2 concentration that has reached at a level of 414 ppm, which seriously affect the health of large population. If we see the last decades from the 1980s to 2010 and 2020, we can see that earlier USA and China, they were contributing the major gases for global warming, and it resulted in the climate change. So our main focus is to protect the earth and environment from this increasing level of CO2. So we are using our own expertise on the microbial enzymes and various nanomaterials and organic solvents, how we can amalgamate simultaneously to products. Right, so effect of elevated CO2 on agriculture and crop yield. The elevated CO2 makes the carbon more available, that we know. Then plant require this carbon resource and also the other resources which are required in higher amount when carbon is present in larger quantity. So elevated CO2 does not directly make these mineral elements more available, but may even decrease the uptake of some element that result in the various problem in the agriculture and total crop that we are expecting. So ability of plants to respond to the elevated CO2 with increased photosynthesis and growth may therefore be limited under conditions of low mineral availability. This effect has greatly affected the nitrogen uptake and assimilation. And if nitrogen uptake is affected, then crop grown with the low amount of nitrogen also show a greater decrease in the protein concentration. If protein content is lower, then the total nutritional requirement that we need from the crops or fruits or plants, that will also be lower. So In CO2 fumigation technologies, we have seen that there is a lower enhancement of biomass production by elevated CO2 under low nutrient conditions. So our motive is how we can decrease this CO2 level from the atmosphere and convert this elevated level of CO2 into some useful products. Then we are using our strategy nanobiocatalysis. So nanobiocatalysis using immobilized enzymes. The nanobiocatalysis, this is the emerging innovation that synergistically integrate advanced nanotechnology and biotechnology and promise exciting advantage of improving enzyme activity, its stability, capability, and engineering performances in bioprocessing applications. And in nanobiocatalysis, enzymes are being attached or incorporated into the nanotextured material, including nanoporous media, nanofibers, carbon nanotubes, nanoparticles that have great efficiency in the manipulation of nanoscale environment of enzyme, and thus promise the exciting advances in many areas of enzyme technology. So how the nanoparticle and enzyme interact with each other and the nanomaterial will affect the activity and stability of enzyme, that we are going to discuss here. Criteria for NBCs, that is nanobiocatalyst. So these are fabricated by immobilizing the enzymes with functional nanomaterial as enzyme carrier and containers. These novel nanocontainers or nanocarriers with advanced hierarchical porous structure for retaining the enzymes can be nanofibers, mesoporous nanocarriers, or nanocages. We can use all these enzymatic system and protein loaded nanoparticle in the nanofertilizers, in the crop enhancement, and in various other fields for the disease detection or drug delivery. So the biological component must retain the substantial biological activity. That is very important in this case. The protein molecule or enzyme that we are loading onto the nanomaterial, that should not affect its original property. That is the first challenge in front of us. And the sensitivity of the enzyme must be preserved after attachment with long-term stability. In case of enzymes, we know that the active center is containing conserved amino acid residue. In case of attachment, if these conserved amino acid residues are making an equivalent bond with the nanostructure, that may result in the loss of the activity, because all the active site amino acid residue that involve in the substrate interaction with the substrate and its hydrolysis or its reaction. So overloading can block the or inactivate the active site of the immobilized enzymes or biomaterial. Therefore, it must be avoided. So an optimized system we need in order to immobilize the enzyme onto nanomaterial. Enzyme of our current interest, that is carbonic anhydrase enzyme. So carbonic anhydrase, this is one of the metalloenzyme that is containing zinc in its active site. It catalyzes the reversible hydration of CO2 into bicarbonate. And this is one of the fastest enzyme known, which has high kcat, that is 10⁴ per second. It is almost found in all organisms, including human beings and microorganisms. So that is a reaction, a reversible reaction, which is being carried out by carbonic anhydrase. So currently my lab is focusing on the conversion of CO2 using a set of enzymes. Major focus is on carbonic anhydrase, FDH, ADH, alcohol dehydrogenase, glutamate dehydrogenase, and formate dehydrogenase. How we can use this cocktail of enzymes or cascade of enzymes to convert the CO2 into some useful products so that we can minimize the level of CO2. Various systems for CO2 conversion. Not only limited to the microbiology or the biological system, we work in the multidisciplinary field, so we can use the materials, chemicals, and biologicals together. We have to identify the particular nanomaterials which may enhance the activity of enzyme, which is suitable for the enzymes. And also, we should have a reaction system, which is the organic solvent or chemical, in which we can submerge our system of enzyme and nanomaterial to catalyze the reactions. Various chemicals or amine-based solvent. Recently, we have published one paper in International Journal of Greenhouse Gas Control, in which amine-based solvent, some sterically hindered amines that can have the inherent property to capture the CO2. The problem with amine is that once they capture the CO2, after some time they get saturated with CO2, and we have to dispose that amine solvent. So what we can do, if we are keeping some biomolecule in that amine solvent and that biomolecule remain stable and remain functional, so that enzyme may capture the CO2 which is already captured by the amine solvent and convert that into some other product. So that is the bicarbonate. So what we did, we merged the immobilized enzyme into the amine solvent, and that amine solvent which is saturated with the CO2, the CO2 is being converted into carbonate. So that was one of the idea. So here in integrated system, we are using the various chemical solvents and porous material like biochar, metal organic frameworks, nanoparticles, nanoflowers, nanomembranes, all that we can use to capture the CO2. So in the preliminary study, we have collected the microbial sample from cow saliva. These ruminant animals, these are the rich source of some bacteria which may carry the carbonic anhydrase. So that carbonic anhydrase is being isolated, and first of all, the cells, the whole cell that we have isolated, that been tested for the CO2 capture capacity. The broken cells or the cell lysate, that is being tested how that can capture the CO2 and convert that into carbonates. So this is the white carbonate powder that we have prepared from the CO2. This CO2 is being converted by these bacteria. These are Corynebacterium flaviscens that we have identified and submitted the sequence to NCBI. Okay, then next strategy to perform the immobilization of enzyme. That is, either we have to modify the sport, we have to modify the nanomaterials to immobilize the enzyme, or we can modify the enzyme itself. This is one of the innovative approach that work I have performed in while working in Huazhong Agriculture University, China. So the hydrolytic proteins like cellulase, the cellulase composed of a, or that comprise of a catalytic domain that is called as cellulose binding domain. This cellulose binding domain, it has the capacity to carry the enzyme interaction with substrate. So the cellulosic substrate will easily interact with the cellulase enzyme, and the interaction base is this catalytic domain, that is cellulose binding domain. If the cellulose binding domain is facilitating the binding of any enzyme molecule with cellulosic material, it means that we can use this cellulosic binding domain to bind the protein with cellulosic sport. So in our study, what we did, we cut this cellulosic binding domain from the cellulase enzyme, not the enzyme actually, but from the gene of the cellulase. The gene for, or the gene sequence for cellulose binding domain, that has been incorporated into the lipase enzyme or some other enzymes that are of our interest. And these enzymes attain the property to get bind to the cellulosic sport. So if we are adding the CBD to any enzymes, that may able to get bind to the cellulosic nanogel. So these are the cellulosic based nanogels that we have prepared from the cellulose by a systematic reduction. So in this case, you can see this is a cellulose binding domain, the whole protein, which is get bind to the lipase enzyme. And this is the fusion protein. In the fusion protein, the main challenge is that we have to retain all the properties of the original enzyme also. Otherwise, if the binding of this domain will affect the active site or will affect the any of the functionality of this protein, then it is also useless. So we have to take care which enzyme can bind to the cellulose binding domain and retain its all the properties. So this fusion protein remain active after binding to the cellulose binding domain. Now this red colored area, this is the CBD, cellulose binding domain. This can easily facilitate the binding of this protein to the cellulosic nanomaterial. So this is a cellulosic nanogel in which the these particles can easily bind to the enzymes, and this is a stable immobilization. There is no leaching or a release of the enzyme in the reaction when we are performing the reaction for biodiesel production or any other reaction system like ester production. So this work was published in Colloid and Surfaces Biointerfaces. One of the innovative approach to immobilize the enzyme. Then in the CO2 conversion, here we can see this is our reaction system where we can add the CO2 from one end and calcium chloride from the other end. And in this propeller, we have immobilized the carbonic anhydrase enzyme, which is working to convert the reversible hydration of this CO2 into calcium carbonate. And we can collect the precipitated form of CO2 in the form of calcium carbonate. that can be the raw material for various industries like cement production plant, in the glass, paper, paint, even in the pharmaceutical industry, such calcium carbonate has been used. So this is the fourth step mechanism of this carbonic anhydrase, which show the deprotonation in the first step, then CO2 addition, followed by ring structure formation, then carbonate substitution. So these are the four step mechanism of carbonic anhydrase, and we published this work in Journal of Cleaner Production in 2020. One of my PhD scholars, she's working on this. So one of a very nice piece of work. And also another nanomaterial for CO2 capture, that is TiO2, that is titanium oxide particles. When we modified these titanium oxide particle with reduced graphene oxide and silver, it has the inherent capacity to convert this CO2 into methanol. So this is the basic scheme how we can prepare the nanocatalyst of silver, TiO2 and RGO. We can use the mangrove leaf extract as the reducing agent because this contain the terpenes and various other reducing agents which can easily reduce the AgCl2, and this Ag nuclei this get incorporated into the graphene sheet, and this graphene sheet is containing silver and TiO2 nanoparticles, and the whole composite become highly active for the capturing of CO2 and converting this into methanol. This work was published in Environmental Pollution, and this is our system. The new idea that is being originated, and we are currently focusing on that. The particles are very interesting. These are titanium oxide particles. And in the enzymatic conversion of CO2 into methanol or carbonates, we need the cascade of the reaction. We need a group of the enzymes. These are alcohol dehydrogenase, glutamate dehydrogenase, or formate dehydrogenase enzyme. So this group of enzyme, all these are coenzyme dependent enzymes. These need the coenzyme NADH. So NADH is being converted into NAD+, and this reduction of coenzyme that is required for the action of this enzyme or functionality of this enzyme. When the reaction is happening, NADH is converted into NAD+. And this coenzyme is one of the expensive product that we need to purchase and we need to add in the reaction system. So once we are adding the coenzyme and this coenzyme is being utilized in the reaction, and once its concentration will over or below the minimum level, the reaction will not work. It will not able to convert into CO2 into further NA product. So what is the new idea here that we are working on? This TiO2 nanoparticle, this has one of very interesting property. In the presence of sunlight or in the photocatalytic reaction, it can have coenzyme regeneration property. The coenzyme which is being utilized by the enzyme, that is being regenerated. So coenzyme regeneration cycle will be maintained by using this nanomaterial. So when the enzymes is being immobilized onto the nanomaterial, that is TiO2 nanoparticle, its coenzymes will never be over, or it can be run for very long time. And when we are using this enzyme and nanomaterial together, the enzyme will convert the CO2 into methanol, and this TiO2 particle will convert the reduced NAD plus into this NADH into NAD plus, and then NAD plus into NADH. So our coenzyme will also remain in the same concentration in the reaction system, and we can efficiently convert this CO2 into methanol. So that's very interesting idea. So this is all the integrated system. We are trying to screen various nanomaterials which can be used to maintain the regeneration of the coenzymes, and then which can efficiently convert the CO2 into methanol and some other products. So we are using the nanomaterials, chemical solvents, even the microalgae. That is one of the new work. One of my new PhD students, she is working on that microalgae, how we can enhance the growth of microalgae using this carbonate, because microalgae is one of very important tool to capture the CO2. And this also require the CO2 in the form of bicarbonates. And our carbonic anhydrase, if it is present in the immobilized form onto nanomaterial, that can provide the carbonates or bicarbonates in higher concentration in the submerged algal system. So there are various nanostructures that we are using for the immobilization of enzymes. These are porous particles, hollow particles, dense particles, doughnut, raspberry, encapsulated, and various our collaborator. We are collaborating with some of the universities and labs in South Korea and China and also in Malaysia. So they are also helping us to synthesize these unique type of structures and immobilize the enzyme onto that and facilitate the conversion of CO2. In our one of the previous work, we have synthesized highly porous SiO2 particles. And the strategy to synthesize this SiO2 particle is that the sole gel of the silica and CNTs, carbon nanotubes, these were mixed together. And after mixing these, we have a composite of CNT SiO2. And this composite is being heated at very high temperature of 700 degrees Celsius or more than 600 degrees Celsius. And all the carbon nanotubes, these were burned totally, and we have the silica left because silica was stable at that temperature. And the porous silica that is containing the channel which are being formed by the carbon nanotubes. So all the carbon nanotubes were burned totally, and we have a very high, this is a schematic presentation. I will show you the original particle in the SAM and TAM diagrams. So this highly porous silica particles, it has the channels and pores, which can be the home for enzyme. And we can merge these particles in the enzyme solution. Enzyme will get incorporate and encapsulate inside the porous particles. Now there is a micro or mini environment is created which can protect the enzyme from direct heat shock, direct chemical attack, or by direct attack of any environmental factor that will affect the enzyme activity. So these are the control particle of silica which are not containing any CNT. So these have been prepared for the comparison with the porous particle. We have seen that after burning of these CNTs, there is a 26 time increase in the total surface area of these particles. That is one of the very good sign. Total surface area to volume ratio was increased so that we can load more protein or more enzyme into this. So in total, it will act as a nano container for the enzymes in which we can immobilize the enzymes or fix the enzymes. Okay, so this is another particle, keratin particles that we have used. The keratin is one of the protein material which is present in the nails, hooves or hairs, wool and feathers of the birds. So what we are working on in another project, the keratin material that is being prepared from the chicken feather biomass, the chicken feather which is which can be available from the poultry waste. The poultry waste is being collected and it is containing, one feather is containing more than 90% of the keratin protein only, and that protein is being extracted from these feathers, and keratin based particles can be prepared for various applications. So many applications of this protein. Even people are using the keratin in the cosmetics for the hair treatment cream and for the preparation of shampoos and soaps also. So anyway, we are using the keratin material or keratin based nanoparticles for the immobilization of enzymes. That is our focus. So we also tried this for the whole cell immobilization, and the whole cells of the isolated bacterium that is being used for the conversion of this CO2, and we find some very good results with this. And the next target is that our students are working on to isolate the enzymes and fix that enzyme onto the keratin based nanoparticles. So these are various parameters for the immobilization. We have to optimize the properties like how much the binding efficiency, how much the immobilization yield and immobilization efficiency of the enzymes. I will not go in detail about all these parameters, but we are discussing about the concepts that we are using. So this is a morphological and elemental analysis of calcium carbonate. After the CO2 is being converted into calcium carbonate, we have to confirm that what type of calcium carbonate. There are various forms of calcite form and some other forms of the calcium carbonate that are being synthesized. What are the actual components of these? So that we can confirm by elemental analysis, by FTIR, by XRD. So all that data just for the confirmation of the product that we are synthesizing, how much the purity of this product and how we can use this in the further application or industry. So in case of CNT SiO2 particle, this is the original particle, which is the highly porous structure. The CNTs were totally burned in this particle. And in this particle, after immobilization of enzyme, we can see this is the nano container which is containing the loaded protein or loaded enzymes. And in case of transmission electron microscopy, we can see this structure, which is also containing the highly porous structure or porous surface, which can be the site for enzyme immobilization. How we can confirm the immobilized enzyme? That is also important or quite important. Here we can see the enzyme is containing amino acid is containing the amine group that is containing nitrogen. So once we confirm the amount of nitrogen in the form of yellow color in the elemental analysis, we can say that this is the reason where enzyme is being immobilized onto the nanomaterial. So one of the nanoparticle of this SiO2 that is being scanned for elemental analysis, and we find the nitrogen content here. That is the enzyme actually. So this is how we can characterize the enzyme after immobilization onto nanomaterial. We can confirm this. These are the time analysis of the same material after immobilization and before immobilization of the enzymes. And another high-end technique is BT analysis that we can do. BT is one of the techniques in which nitrogen absorption and desorption theorem we can use. These are nitrogen desorption isotherms. In that case, we can analyze the total BT surface area. So before immobilization, the BT surface area was 494 meters square per gram, and pore volume was 1.3. Pore diameter was 11.95. But as we immobilize the enzyme, so in this technique, we can confirm that if BT surface area is decreasing, it means that we can quantify the amount of enzyme immobilized. The pore volume was decreased from 1.3 to 0.3. The surface area was decreased from 494 to 170. It means that some of the area was occupied by enzymes. So that is the one of very good technique to confirm that how much the enzyme was immobilized into the particles, how much the pore diameter was decreased. And so these are the some testing enzymes that we have immobilized, like how much the lipase, how much the HRP, phosphoradis peroxidase, how much the lacase was immobilized onto this. So onto the porous particle and dense particle, we have immobilized and confirmed by BET surface analysis. So this is the just graph for the BET analysis, how much the pore diameter, how it has been decreased after immobilization. And this work was published in Chemical Engineering in 2018. Various other instrumental methods like FTIR, circular dichroism, confocal microscopy that has been used to characterize the enzyme onto nanomaterial after immobilization. So using this fluorescent dye, FITC fluorescent fluorothiocyanate, that can be used to locate the enzyme, because this is protein binding dye which can bind only to a particular amino acid in the protein. It will not bind to the control particle. So we can also confirm the immobilization of enzyme by this FITC labeling. And how much the change in the secondary structure of enzyme, that can be confirmed by CD analysis, circular dichroism. After immobilization, if there is some change in the 3D structure of enzyme, we can know that this much will be the change in alpha sheets or alpha helix and beta sheets. Moreover, this is another result of the contact angle analysis, in which the hydrophobic sport, the CNTs were hydrophobic in nature.
Please unmute participants. Please unmute yourself.
Yeah, now it's fine. There is some background noise. It's fine. So contact angle analysis to measure the hydrophobicity or hydrophobic property of the nanomaterial that we are using. The CNTs, they have created some hydrophobic environment. And in enzyme, if we see the 3D structure of enzyme, the lipase particularly, that enzyme has the lid opening mechanism. The lid is containing some hydrophobic amino acid residue. So in the hydrophobic environment, why the activity of enzyme is being increased? That is due to the hydrophobic property of the enzyme, because the hydrophobicity of the sport, that will repel the hydrophobic lid of the enzyme. The lid is always on the pocket of the lipase enzyme. If that repel the lid will open, the substrate will easily enter into the active site of the enzyme, and it result in the increase in the activity of enzyme. So that is also one of very good idea, that how we can improve the activity of enzyme by selecting a particular immobilization sport. If our emulsion sport is like hydrophobic, so a particular enzyme with some hydrophobic lid can enhance its activity. So these are some new ideas about increasing the activity of the enzyme. So these are the thermostability and reusability parameter of immobilized enzyme. The enzyme has retained its activity even after 10 to 12 cycles of its use. We have used this in the reaction system, so it remains stable for a longer time. And this is a biosensor that has been developed by using the lipase onto this SiO2 porous material and various parameters for the degradation of these fatty acid and tributylene substrate that has been um, analyzed. So we can use this this biosensor for the detection of fatty acid in the blood, in the food sample, or in various other products that we want to analyze how much the fatty acids are there or how much the cholesterol was there, that we can use. Even for the cholesterol detection, the cholesterol oxidase enzyme that can be immobilized onto the such nanomaterial or such nanoparticles. So all these are the electrochemical analysis for the lipase biosensor. The work was published in Chemical Engineering Journal. Another interesting particle is magnetic core shell SiO2 particles for the emulation of various enzymes. So this particle is again very interesting particle, which is containing a composite of nickel oxide and V2O5 with SiO2. The core is containing Ni3V28 and SiO2 is the covering of this. So this has the magnetic property, and we can immobilize the enzyme molecule onto the surface of this and inner side of the particles. And after immobilization, we can cross-link with some cross-linking agent like glutaldehyde or formaldehyde, which make a network of the enzyme onto the surface and inner core of the particle, and it become a highly active nano container. So any protein molecule or any enzyme we can fix into this particle, and we can use this for commercial application or industrial applications. So all these are the results of transmission electron microscopy and elemental analysis for the free and immobilized enzymes. Then various enzymes like lipase, glucose oxidase, HRP, and lacase that we have used for the immobilization onto this NiOSiO2 particle, and the short form that we are using for this, not the complete name. So the activity, the immobilization yield and efficiency that we have tested, and for various enzymes, it is showing that for lacase, it is around 97% of emulsion efficiency. So we can go with the best one, like lipase has the 241 emulsion efficiency. And also at the same time, we have to check the emulsion yield, how much that is, how much the activity it has retained. So if all the parameters are suitable for our applications, the activity, stability, efficiency, and protein loading, all these are okay for our work, then we can go with that enzyme. So here we forwarded the work with lacase enzyme. Lacase has been immobilized onto these particles, and it has been used for the various applications like degradation of the lignin content and phenolic compound in the biomass, because phenolic compound we need to remove during the synthesis of the biofuels. So this is the original structure of this particle that we have, and after immobilization it looks like this. This is the scanning electron microscopy. This is the original particle without an enzyme. After immobilization, we saw that all these pores and all this surface area that has been covered. The inner core was empty, that is being loaded with the enzymes. After that, we can cross-link with this with the glutaldehyde. So these are the elemental analysis results. All these are for the confirmation of enzymes. So here you can see the green color nitrogen content. That is the symbol of amine group. Amine group present in the amino acid. Amino acid are present in the protein or enzyme. So we confirm the presence of enzyme onto the nanoparticles. All the CD analysis and F5TC, I will not go into the detail of all this data, because I have explained why these techniques are important. So BT analysis, again, we have done for the immobilized enzymes for this. So we can see that for the same particle, the BT surface area that was before immobilization, it was 233. After immobilization, it was 127 meters square per gram. So it was decreased. So it means that enzyme was successfully loaded or immobilized into the particles. All these are the XPS result for the confirmation of the functional groups and enzymes onto the nanoparticles. Okay, just let me skip all these. So temperature and pH optima, we can see that the free lacase at various temperatures like 50, 60, 70 degrees Celsius, we can see that there is a direct decrease in the or sharp decrease in the activity of the enzyme. This is the relative activity. While in case of immobilized one, we can see here there is a both preparation, cross-linked and free one. Both are retaining their activity for the longer time. So even at higher temperature, so at 70 degrees Celsius, immobilized enzyme was stable because enzyme was protected from the direct heat shock. And the thermolabile amino acid residue which are present in the enzymes, these are not being harmed by the increase in temperature. Mostly the thermostable amino acid residue like aromatic amino acid residue, these remain exposed to the heat shock and they can tolerate the higher temperature also. And in case of pH, there is a continuous decrease in the free as well as immobilized enzymes. So it was not useful for the wide range of pH. We cannot use in the different ranges, but in the acidic conditions in the 3, 4, 5, all the enzymes were stable. So mainly we can use this in the biomass degradation. That is the stability at different temperatures at 35, 45, and 55. We can see the immobilized one was stable for the long time when we have incubated the enzyme in a water bath for various time periods. So up to 300 minutes that we have analyzed the result, the immobilized one was retaining its activity and stability. This is the pH stability, which was also better for the immobilized one as compared to free one. And then the reusability data, we can see up to 12 cycle of reuse, we can reuse the immobilized one again and again. So we can perform one reaction, we can take out the enzyme by centrifugation, and we can put this in the fresh reaction system, and we can use this again. So up to 12 cycle, it has almost more than 80% of activity, which is around 85 or 86% of its original one. So that's why this preparation was also very useful. Then we applied this for the degradation of all these phenolic compounds which are present in the plant biomass, that is comaric acid, gallic acid, ferulic acid, tannic acid, and we have seen the hydrolysis or degradation or conversion of these phenolic compounds. So various CV analysis, cyclic voltammetry that have been used to measure the redox potential during the conversion of these phenolic compounds. For tannic acid and ferulic acid, they have a different rate of conversion. It is based upon the complex structure or complexity of all these phenolic compounds. Like we have the tannic acid, which has the very complex structure. So it will take much longer time as compared to another ones. So that's all the highlights of our work that we are working on. And so we are focusing on microbial system, enzymes, and materials. So we published some of the books in Springer, like Advances in Carbon Capture Utilization, Chemobiological System for CO2 Utilization, Nanomaterial for Biocatalysis. So these are the recent books in last few years. And also about the biopolymeric nanomaterials, keratin, lignin, exopolysaccharide, then nanosensors for the smart agriculture, this plant microbial interaction with smart agriculture biotechnology, and all these in paper and wood engineering. So that's all about the various papers that we have published, which are available on the Google also in our profile. If someone interested, may can visit. Then finally, beside this, there are various applications on the immobilized enzyme in the industrial processes. Depending upon the type of enzyme, its molecular weight, chemical nature, nanostructure for the immobilization, that can be prepared. What type of nanostructure we need, we should know first the property of that enzyme. Shape, size, and other property of the nanotexture surface can be tuned according to the need of application. So that is the conclusive remarks of our presentation, and we are open for the collaboration with the researcher who have expertise in the novel nanostructures to develop the efficient biocatalyst, to develop the new system for CO2 capture and environmental protection. After all, we have to protect our society, we have to protect the human beings from all the environmental disaster like climate change and global warming. So that's what our focus is. And I would like to acknowledge all my previous affiliation and present affiliation. Like I worked in, I did my PhD from Himachal Pradesh University. Then I moved to Huajang Agriculture University, Konkuk University, Korea University, and some funding from ICMR also. Currently, we are working on GOIT, JP University of Information Technology. So I'm thankfully acknowledging all these. This is my contact, and phone number. One very young group of researchers, some PhD student and master student, they are working with me. So we are just a very small group who are working here. So with this all, I would like to thank you again, organizing committee and all the members for inviting me for this lecture. I hope that it is helpful for audience. So thank you very much.
Thank you very much, Dr. Ashok. It's a very nice presentation, and you are really working on a very important aspect, that is global warming and CO2. Like increasing CO2 is a major problem nowadays. But CO2 is also a renewable source, and you are converting CO2 into some usable form, that is calcium carbonate. You developed some good technologies, and another aspect you are working is very interesting, that in India emulation is very important aspect, because emulation sometimes we lose the activity of enzyme, but you develop some nanoparticles that have like that is not hampering the activity of those enzymes. In fact, it is like making those enzymes more robust, what they have in the in their native form. So yeah, it's a good piece of work, and it is we can see that it's already reflecting in your publication. So I congratulate you again, and yeah, I will be very happy to visit your institute sometimes, and I'm really happy to collaborate with you as well. Okay.
Yeah, you are all always welcome here. This is one of very beautiful place and near to Shimla also.
Yeah,
So-
I was there for I think few years back. There was a conference in your campus. I think at the time you were not there. You are in, I think, China or in India.
Okay, okay.
Thank you, Dr. Nudda.
Yeah, very good
Excellent
afternoon,
talk.
sir. Thank you very much.
Excellent talk.
Yeah, thank
I
you very
hope
much.
there are questions from the audience also.
Yeah, sure.
Any questions? In the chat box, there is one question from Dr. Sevanay. Which are the microbial organisms release carbon dioxide or almost all types of microbes?
Okay, sir, which are the microbial organisms release more CO2 or almost all type of microbes? Microbes can be aerobic and anaerobic in nature. Some of the microbes, they may utilize the CO2, and some of various other microbes which are aerobic in nature, they are utilizing the oxygen for their growth. So maybe your question is, I don't know in which context it is, but my answer is only they release more CO2, and almost all type of microbes. Not all the microbes, they will release the CO2. Some of the microbes which are anaerobic in nature, they will utilize the CO2 for their growth. Mostly when, if you see the pond ecosystem, in the lowest bottom of the pond, we can find in the mud, we can find the anaerobic organisms, which are growing at the cost of dead and decaying organic matter. They are hydrolyzing that. Maybe most of the archaea or methanogens, they are utilizing the CO2 or methanes for their growth. So that's my explanation for this.
I think there's another question. I can't really get this.
The nanoparticle.
The nanoparticle which inhibit the iron process in the rice crop.
Iron chlorosis in rice crop. I'm sorry, because this is just out of context of my area. So I will not be able to answer this iron chlorosis in the rice crop particularly. Maybe in future, maybe I will try to search for this question, or professor or any audience from the audience if want to answer about this, because I don't have any.
Iron chlorosis is due to deficiency of iron. So maybe iron nanoparticle may be the solution for that, because sometimes iron chlorosis may also occur by the magnesium. So we have to have some iron or magnesium nanoparticle. They could be helpful in reduction of the iron chlorosis. So I think Dr. Nadha, I'm really happy to see you, and I talked many times on the phone. And
yeah, thank you, sir. Thank you for inviting me.
You're working on a very good area of global warming. My question is, when you are using calcium chloride and converting into calcium carbonate. So this is not an expensive affair because calcium chloride is also a costly raw material. You have to have some cheaper sources so that
Yeah, actually, in that case, we are using in very low amount calcium carbonate. Majorly, our focus is the large amount of CO2 that is being captured. So it was the previous study in which we have used the calcium chloride also. Now we are directly focusing on the conversion of CO2 into methanol, in which we don't need any calcium chloride to be used. Here, the set of enzyme and the nanomaterial that TiO2 based nano composite with reduced graphene oxide that can be used for the conversion of this CO2 into methanol without the use of calcium chloride also.
have you tried for upscaling of your technology and answered how much CO2 can be sequestered by the technology which you are developing using immobilized enzymes?
No, still we didn't perform the upscaling. Only at the lab scale we did with the minute amount of enzyme that we have prepared, because the purification of the enzyme from the microorganism, that is one of the tedious process. So our students, they are working for last 2-3 years on the procurement or purification of enzyme on large amount, which can be efficient for the conversion of this CO2. So upscaling is the maybe our next project that we can work on. And also one of the important way we can use this summer system for the microalgal enhancement. So that is one of very good idea where we can increase the growth of microalgae, and microalgae simultaneously will capture the CO2. And the CO2 which is already dissolved in the water or in the contaminated industrial water, that can be converted by this carbonic anhydrase into bicarbonates. And these bicarbonates are being absorbed by microalgae, and its growth or its biomass will enhance. So if biomass will enhance, it will capture more and more CO2. So that is one of our project we are currently working on.
So later on, algae could be utilized for biofuel production, etc.
Even that algae, if that is proteinaceous in nature, we can use that for the purification of other proteins for tablet preparation and in the pharmaceutical industry. And if there is some lipid content is higher, we can use that lipid content for biofuel production. So various application of these microalgae depend upon which species we are going to grow, chlorella or some others. So that is one of our project.
Okay, very nice, very nice. Keep it up.
Yeah, thank you very much, sir. So I would like to invite maybe you people sometime whenever you get the chance to visit JOIT campus, and we can have interaction physically, and we can start the collaboration.
Thank you very much.
Yeah, thank you, sir.
You are mute, sir. Bhavla sir, you are mute.
Now I request Dr. Anil Kumar, Director of Education, to facilitate when eminent speaker from with a moment.
Okay, Dr. Ashok Dadda, we are delighted to have your presence in this webinar. And hopefully this university, whenever need your help, because we are newly developing university. So colleagues of mine, if they really require your help, try to provide so that some of the work on this important technology would also be initiated in this newly developing university. Thank you.
Yeah, thank you very much, sir. Thank you.
Thank you, Dr. Munda.
Yeah, thank you, sir.
Now our next speaker of the day is Dr. Vijay Kumar. Dr. Vijay Kumar, are you there? Yes. Most welcome to you. Dr. Vijay Kumar obtained his PhD in 2006 from PNAU and postdoc on nanoscience in National Chemical Laboratory, Pune, on photothermal and sensor application in Taiwan. He has been visiting scientists in NIOSH, USA, and have a couple of patents and few awards such as Outstanding Reviewer Award in elsewhere and finalist in Swiss Young Researcher. Currently, he is working in Institute of Nanoscience and Technology, Mohali, and have introduced the concept of targeted agricultural inputs possible with nanotechnology. He has have 1,500 citations with over 30 publications on this. Over half of the impact, among them are more than 10 impact factors. So, Dr. Vijay Kumar, I welcome you and welcome you to present your talk on biomaterial-based innovations for plant protection and growth.
Yeah, is it visible?
Yeah, it's.
Is it full screen now?
No. No, now it's full screen.
Yeah. Okay. So shall I start?
Yes, please.
Right. Yeah. Very good afternoon to everyone. Before going to the talk, I wish to thank Dr. Prashant Jambulkar for giving a kind introduction about me. Just hold on, my intercom is ringing. I'll just attend it. Excuse me. Hello. Neha, no, I'm on meeting. I will call you later. Thank you, Mika. Bye. Yeah, okay, bye. Yeah, sorry. Anyhow, it will be objecting. That's why I attended. Sorry. Yeah, so I wish to extend my gratitude to the chief guest, Dr. T.R. Sharma sir, DDG. Like he is a great inspiration and motivation for me since the time he was in Nabi here in Mohali. And I extend my thanks to the chief patron, Dr. Arvind Kumar sir, and the organizing secretary member, Dr. Ambrish Chandra, who's also a partner institute and patron of this event, and Dr. Anil Kumar for inviting me to deliver a talk here in your conference on nano size, big impact, nano revolution for transferring agriculture, food and nutrition. And actually, Dr. Jambulkar was kind enough to give me a nice slot. But anyhow, because I was we had another visitor, ACS editor was visiting our institute, and so it was once again it was related to CO2 sequestration and other things which Dr. Nada was presenting just before. So that forced me to take this slot. And I don't know, many of you may be tired by now, having heard so many talks. So just deliberately, I have put this slide for you. Thank you. So that you all feel very glad that this talk is going to end. But anyhow, just bear with me for next 20 minutes. I'll not I'll not extend for 40 minutes as per the plan. I'll try to give some kind of a juice of what we are doing in 20 minutes, and then let's stop there. Otherwise, I think it may be not a good idea to continue that way. Okay, so the outline of the talk is a brief introduction where I don't want to skip anything. Okay, since because we don't have time, I don't want to skip anything. But I'll give a brief idea. Okay, I'll say small thing about our institute and what is Nano, and then what we are doing outside, and then specifically this talk that is covering the carbon nanoparticles, and then we have developed some 5 different technologies that have been appreciated, well taken, and some of them have a future to take it to the field level, especially some are with company collaboration, and so most of them we got it published in ACS Applied Material Interface and Conclusion in the acknowledgement. Okay, so this is our institute. So our institute was started in 2013, and our first recruitment happened in 2014, and we were like 32 faculties. 20 faculties were hired, and then subsequently another couple of recruitments happened, and now we are full house with around 36 faculties. This is our old campus, transit campus, where we were running through hiring the building, and then like now very recently in January this year, we have moved to this very beautiful glass building that you're seeing, magnificent and gigantic. So what we are working is like our institute focus on, broadly can be classified as biology, chemistry, and physics. In case of biology, it is agriculture, therapeutics, and chemical biology things, and sensor diagnostics. In case of chemistry, it is environment and soft nanostructures, and in case of physics, some device development and other things. Of course, we do collaborate with many things, and as like you'll see in the next slides. Okay, so in nanotechnology, one is top down and bottom up. These are all the things I think Dr. Anil Kumar sir have already discussed with it. So I'll like skip this. And yeah, what is nano? This part I want to like emphasize, because this will be good to know, so that like the students especially gets motivated. Okay, this is especially for the students. Okay, let me take like little time in this slide. Okay, so when you confine the particle, okay, so you are like breaking like this. Okay, I hope you all are able to see my mouse. Okay, so you are reducing the size. Okay, once you are reducing the size, you are exposing the surface. Okay, so when you are exposing the surface, automatically the reactivity increases, and the delivery or whatever that we are doing with the plant is all increasing. Okay, apart from that, there is a very interesting phenomenon in nano. When you're reducing the size, okay, say for the same material that is existing in the periodic table, the same material gives a different property. Okay, that is very interesting. Say for example, when you take gold is yellow color. I will explain that in the next slide. Okay, and when you reduce the size, it gives a different color. Okay, so this is completely unknown phenomenon that was known before. Okay, and the same way magnetic property, when you're reducing the particle size, you happen to see something called super paramagnetic material. Okay, so the space, what is the beauty of this? Okay, it gets attracted to the magnet. Okay, at the same time, when you remove the magnet, it immediately goes back to the colloidal state. Okay, it don't stay together. Okay, so these are all the interesting part of the nano. Like say for example, the carbon, which is known to be non-conductive, when it is reduced to nano size, confined, it shows conductivity, high conductivity. Okay, as much as copper or anything else can do. Okay, these are all the interesting properties that they have, and that we can tune it for our application. Okay, say for example here, when gold is like this, okay, light cannot pass through it. Okay, when you're confining it, okay, it gets accommodated, it becomes nano size, and the visible radiation that we are having is 300 to 700 nanometer approximately, and these are all bigger than the nanoparticle that you have synthesized. Okay, so the particle gets accommodated in the wave. Okay, so the light wave is nothing but electromagnetic radiation. Okay, so it is an oscillation of charges. Okay, so when the oscillation of charges happens, and when your particle is confined in that wavelength, the electrons on the particle, okay, that comes as a plasmon that gets projected away from the physical body of the material, okay, that gets polarized by the charge here. Okay, so this charge starts oscillating with the charge of oscillation of this wave. Okay, so that oscillation of this material is a characteristic of the material size and shape. Okay, so if it is a gold nanoparticle at 13 nanometers, it will absorb at a frequency of 520 nanometers because it oscillates in the frequency of 520 nanometers. Okay, so what is the use of it? Whatever is absorbed have to deliver as heat. Okay, so here what we do, some of the polymers can hold the drug with the particle, okay, and then you allow it to circulate and it gets accumulated. And here, for example, it is accumulated in the sternum region and also in another region. Here is what we need the treatment. So we shine the laser here, and then the polymer, the thermosensitive polymer, because of the heating, it ruptures. Okay, this heat that is generated by the laser will be very mild because the body doesn't have an absorbance there, only the particle have the absorbance. Okay, in this case, like we have done very nice work here, which like since because of point of time I'll skip it here. Okay, so what we do in our lab, reference to agriculture. Okay, so in agriculture, these are all the 4 major steps, that is production, protection, preservation, and processing. Okay, so in all these 4 cases, we try to concentrate. Okay, say for example, in production, of course, fertilizer is important. So to deliver fertilizer, we have used these kind of mesoporous silica. In this mesoporous silica, you'll have 2 to 5 nanometer of pores. This green one are the pores. Okay, this pore size can be tuned based on the mycel that you select, and you can load it with fertilizer, and then this is covered with the ketone or as any other sensitive polymers, and then whenever the root comes nearby, the polymer swells and opens, and whenever the the root doesn't come nearby, okay, the polymer stays as such. Okay, it is compressed. Okay, so the one is here in a targeted fashion, it is releasing the nutrient. Okay, and here in a targeted fashion, it is not opening the pore. Okay, so it doesn't allow the soil solution to do any other undesired activity to convert the phyto-available fertilizer that you have to convert into phyto-unavailable form. Okay, so it is doing 2 functions. Okay, one is it is releasing in a targeted fashion. The other one is disclosing and protecting the protein from the, fertilizer from converting into phyto-unavailable form. Okay, so coming to the protection. Okay, protection, we have done extensively some of the work. Okay, this we'll be dealing in depth, so let us skip it here. Okay, and then coming to the preservation, this also I'll explain you, because this comes under the same carbon-based system that we have developed. And then processing. In case of processing, we know in agriculture, this mushroom is a very nice vitamin D. It has a huge amount of vitamin D precursor, that is ergosterol. Okay, so this ergosterol can convert to this active vitamin D only when the cleavage of this C9, C10 bond happens. Okay, this is in the B ring of this ergosterol. Okay, so this cleavage will happen only when the UV light is there. Okay, so for this in US already people are using UV light, and then the retail you get one mushroom that is illuminated with UV and the other mushroom without the illumination of UV. Okay, so here what we have done is, you may have heard about fluorescence. Okay, in fluorescence always what happens, it takes higher energy light and then it gives lower energy. Okay, this is because the triplet state that is happening just below the excited state. Okay, there is a loss of energy in between. Okay, so you get a lower energy conversion. Okay, here this is a kind of up conversion material, interesting material that can up convert than the down convert, than the conventional down conversion process. Unlike the down conversion conventional process, here in the up conversion nanoparticle, NIR radiation can be taken and higher energy UV light can be given. Okay, so we have made a disc like this, okay, of this up conversion nanoparticle, and then we inserted in the mushroom and we were able to convert this in the solar radiation. So you get the mushroom enriched with vitamin D by 3 times. Okay, these are all the things that we do. And then we do in all the possible areas of production, protection, preservation, and processing, we do develop some sensing process with the electrochemistry, same resistor, and also with the optical nanoparticles. So coming to the carbon, okay, how do it synthesize? Okay, in carbon, in case of carbon, one basic thing that one should know is there are two types of carbon. One is sp3 carbon and the other one is sp2 carbon. This is simply because of the different way of carbon-carbon hybridization. Okay, when the carbon is having 4 other neighbors, it is called sp3 carbon, that is diamond-like carbon, like here. Okay, and the other one is sp2 when the carbon is having only 3 other neighbors. Okay, that is sp3 carbon, that is graphitic carbon, which can be in different allotropes. Say for example, fullerene here, carbon nanotubes. Of course, Dr. Nata was speaking about carbon nanotubes in the previous talk. Okay, so apart from this, there is one more allotrope, that is graphene oxide, which is the cheapest form of nanocarbon material. Okay, so how do this can be synthesized? You just have one. This is very simple. Okay, this is 1 lakh rupees of this tubular furnace. One should have. You just have this in biomass. Keep any biomass inside it, and then you run argon or nitrogen in it, and then you heat it to above 600 degrees. Then you'll get a different graphitization that is happening there. Okay, based on the degree temperature that you give. Okay, so graphitization is nothing but the carbon, this sp3, I mean sp2 carbon, which I said, that forms as a sheet like this. Okay, and then the sheet is arranged as a bulk carbon. Okay, that we see in the coke in our home. Okay, so what we do, we put it in the very strong reducing, I mean oxidizing agent H2SO4 and KMNO4 that form a in situ radical that oxidizes and breaks the bond between the strong carbon-carbon bond and then tangles outside. That makes the zippering happen, and then the sheets come out. Okay, exfoliation happens. Okay, and then you can slightly add a reducing agent and you get the reduced carbon, graphene oxide. Okay, so coming to the pesticide now. What is the problem in pesticide? Okay, the pesticide 2.7 times it is going to increase by 2030. Okay, already we are applying huge amount of pesticide. This is because of the huge population that is going to be this planet is going to support. Okay, and all these pesticide that is being applied is going to a huge loss, as you are seeing on the right side schematic. Let me skip it. Okay, so basically whatever that you are applying completely gets washed away, as you can see in the left side panel. Okay, whereas here you see this red sheet. Okay, so this carbon that we have prepared, as I said, it is a sheet. You are exfoliating it and then it is a sheet, right? And that you are applying on the leaf, and even if water comes and flows, the sheet will not be going along with water, unlike the emulsion, conventional emulsion that is there. Okay, this like one is sheet. Okay, apart from this, there is a lot of chemistry on the surface. Okay, it have a high binding ability, and it is having high surface area that is very energetic. Okay, to bind with anything that is coming across. Okay, imagine 1 gram of graphene oxide have 2,600 meter square surface area. Okay, just 1 gram of this graphene oxide will have 2,600 meter square of surface area. Just imagine it, how voluminous it is. So here what we do, we apply it. Okay, we take this graphene oxide and then we have this chloropyrifos model pesticide, and then we apply it to the leaf, and then we give a drift, and then we were able to see that unlike the emulsions, these graphene oxide is able to hold on the leaf, and so the drift loss is reduced. Okay, so different way of targeting. Okay, targeting in case of animal system, you are using folic acid based or the laser you are using. Okay, here in the case you are reducing the drift. Okay, when you are reducing the drift, you are increasing the target by 40%, because the 40% is the statistics that is given approximately of the pesticide loss, drift loss that is happening. Okay, and then you are substituting the micron composite, composite that is formed with the emulsion. The emulsion, the minimum size that you can get is 200 nanometers. 200 micrometers. Okay. 2 micrometers. Okay. So this you are dispersing into nano size. So automatically the dispersion increases the statistics of hitting the best. Okay, so there you are increasing the target. Okay, and then the persistence is there. Okay, when it is persistent in the leaf, that again causes non-targeted effect. So what we do here, we decorate some semiconducting nanoparticle, cheap semiconducting nanoparticle. Okay, so that will cause the degradation or reduce the half-life of the pesticide. Here we have optimized the concentration of this semiconducting particle in such a way that the half-life of the pesticide exists more than 15 days. Okay, and this can again be controlled based on the dispersion of the graphene. Okay, this is all the characterization of the material, which I don't want to spend here. And here the interesting part is the semiconducting particle that we have chosen have 2 absorbents, as you can see here. One is at NIR and the other one is UV, visible. Okay, the visible one helps in the degradation of the pesticide, whereas the NIR one helps in the heating. As I mentioned, in the human system, the photothermal release in the drug happens. Here the photothermal pesticide release happens. That is how the targeted release system have been given. Let me skip all these things because it's a want of time and I should not bother the students. I think whatever the time I have given already, I'm reaching 20 minutes. I'll try to do it, okay, as much as possible.
No problem.
Yeah. Okay. So the second part is triple smart material, okay, that we have developed. Okay. This part, let me thank Dr. Vinod, okay, one of my friends. Who was giving, I think he gave the second talk today, right? So he was talking a lot about ketosine. Okay, he exposed ketosine much the same way. This ketosine is very well explored for its acid-sensitive opening ability. Okay, and it releases the drug that you're coating, and then it helps in the targeted. Targeted control of the disease. Okay, so in the same way here for the anthracnose, we have loaded Captain, which is highly hydrophilic, can be easily washed away, unlike in the normal formulation. Here, since because you are using it with the graphene oxide, since I said this, it can be stably standing on the leaf. In spite of being hydrophilic, this Captain can stay much more better than the regular formulation with the carbon. Okay. And here again, this copper selenide is there, and copper have a very good antifungal property. So it gives complementary effect in the
फ्रेंड्स ग्लाइफोसेट और नहीं आ रही है अपनी ग्लाइफोसेट
राजवीर सिंह जी
नहीं आ नहीं रही अब आ नहीं
म्यूट
रही
कीजिए
और क्या नहीं क्योंकि जो ढांचा
राजवीर
पलट
सिंह जी
उसमें तो डलेगी
Yeah, that's okay, I think. Okay. Yeah, so one smart behavior. So triple smart here we have explained. First smart behavior is it binds to the leaf. Okay. And second smart behavior is it is ketosine. So when the fungal spore starts like upper sodium and other things, the acidity increases and it releases the Captain and helps in the control of the fungus. Okay. And the third smart behavior here we have explained this often. Although it is graphene oxide, it controls the drift loss, but still the loss is not completely stopped. Okay, still there are significant amount of loss by washing. Okay, so that washing, what happens to the soil? Okay, especially with Captain. Okay, Captain is not a very recalcitrant material, but still Captain is highly hydrophilic. When it is washed away to the ground, immediately it goes to the groundwater, and in groundwater its half-life is more. Whereas in soil, its half-life is very poor. Okay, so we have to help the Captain to stay in the soil for easy eco-friendly degradation. Okay, so what happens is This ketosine in the soil, because of the carbonate and other ions present, it is not able to release the captain. Okay, so we are explaining three smart behavior. One is holding on the leaf, other one is smartly releasing to the fungus, and the third one is it is holding the groundwater, I mean soil, and unlike leaving it to the groundwater and having a half-life. Okay, so this is again all these characterization about the material, and then this is about the smart behavior that we have explained. One is adhesion, okay, and the ability to hold from leaching, okay, and this is tested with the like chili, and we were able to see that the spot by the anthracnose is being reduced in the formulation. Okay. So now coming to the third protection, okay, like this pesticide is always like hard to convince, okay, because now more of eco-friendly passion is coming or fashion is coming. So it is always better to control by pheromones, okay. So here what we have done is we have made a formulation for pheromone. So this is a nice story. I feel like somebody just for you people to open up. See, this is a Nobel Prize that is given in 1973. Okay, what they have seen is the social behavior. Okay, when the duck, geese hatches out, okay, from they starts immediately walking behind the mother. Okay, if the mother is seduced, they starts walking behind any dummy object. Okay, this was the observation by this Nobel scientists. Okay, so basically, this social behavior exists by genetics and not by teaching. Okay, and pheromone is one like that. Okay, and this is coming by generation. Okay, so this is a pheromone for the Tuta absoluta, which is tomato pin. Okay, so the challenge in pheromone is it is more than one compound that should be released in the same ratio as it is given here. Okay, so we made a graphene oxide. I mean, and before this, these are all the formulations that are there. One is this kind of release mechanism where you have a thread and it slowly releases, or otherwise you load it with the cellulose paper and then with a commercial septa, which is a rubber. Okay, rubber have its own problem. Many things are there. So we have developed this graphene oxide based system. So the graphene oxide forms a pile like this that will be able to release slowly by extending its diffusion path. Okay, so the release kinetics is inversely proportional to the diffusion path. So in this material, what we are doing is increasing the diffusion path. Okay, let me skip all these things. And coming to the production part. Okay, so this graphene oxide, based on the reduction that oxidation that you are doing, it can form two like forms. One is semiconducting property it can show, okay. Other one is can show conducting property, okay. And always this graphene oxide is heterogeneous, okay. So this we thought, why don't we use this to manipulate the chloroplast, okay. So in the chloroplast, we have added the graphene oxide that is heterogeneous, and here we have reduced the size of the particle so that it becomes semi-conducting. Okay, so it takes light and gives electron for the photosynthesis apparatus to function. Okay, so that is one way of stimulation to the chloroplast. And the other way is it also see only phycocyanin is the only redox carrier there. Okay, so this scatters only 15 to 25% in the plant. Okay, so this graphene oxide is also having the potential act as a redox event, okay, because of its functional group. So that is also able to increase the chloroplast activity here. Okay, so let me skip all these things. And this is coming to the preservative. Okay, so preservative, so generally it is applied directly on the fruit. Okay, so that will cause carcinogenic effect in the long term. Okay, chronic effect can be caused. Okay, so here, and most of the time the conventional preservative that is used is sodium sulfite. Okay, this sulfide is being released slowly because of the vapor pressure increase that enters into the fruit, and it starts converting the quinone into quinone sulfide. Okay, so this quinone is the main part in the fruit that is helping in the health improvement. Okay, once you convert it to quinone sulfide, the phenol oxidase will not be able to act on it. So lignin formation will not be there. So the browning will not be there in the fruit. But the problem is you have already converted the quinone to unhealthy quinone sulfide. Although it is not showing browning, you are already converting into unhealthy quinone sulfide. Okay, so here what we have done is we have used some antioxidant on the graphene oxide, and then this have been casted as a paper. Okay, and then that has been inserted into the punch, and we were able to extend the shelf life of banana. Okay, and this is another interesting work. So for the gene manipulation, so this is simple. Okay, by older biosynthesis process, you have the fungus, and then the fungus is taken with the gold chloride solution, and the gold nanoparticle is synthesized in the fungal body intracellularly, and then you heat it in the tubular furnace, which I showed in the initial stage, and you get this carbon with the gold nanoparticles in it. Okay, this you use it for the gene gun based plant genetic engineering instead of the micron sized gold particle that is given by Biorad. And we were able to increase the gene manipulation by 4 times, and the transformation and the regeneration have been increased. Okay, we have given some of the reasons why this has happened. And yeah, this part I have already explained to you. So I think yesterday itself, Dr. Ramesh Ralia have also put up this slide. It is all these conferences, we have to look into this, because DBT have given a very nice guideline how to take these nanoparticle technology to the agriculture application at the field level. Okay, so this is the conclusion of the talk. Okay, if anybody is have slept, they can now wake up. I will give a quick recap of the complete talk that I have given. So it is all about graphene oxide. Okay, one is targeted pesticide application. For targeted pesticide application, diurnal pest, which actively feeds in the daytime, we need photothermal conversion. Okay, this material gives photothermal conversion. And for the preservative loading, we need a huge amount of functional groups. Okay, this material gives 60% functional groups. Okay, no other material in this world can give 60% atom of functional groups. Okay, and with the photosynthesis, it has both semiconducting property and conducting property based on its heterogeneity. Okay, so it is able to improve the photosynthesis. Okay, and the drift loss, I explained that it is having high flat surface with high energy, so it is able to bind to the leaf, reduce the drift. Okay, and then it is forming a stake. Okay, so that is able to increase the pathway of diffusion that reduces the release rate. Okay, and as a token of appreciation for all the nice work that we have done, we have been given a chance to write Progress in Material Science. This is a very prestigious journal for writing a review of 40 impact. So we contributed in it. So anybody needs any more detail of ours, as well as many of the counterparts globally available, you can check this nice review. Now we do some outreach as well, and this is my research group, and thank you.
Thank you, Dr. Vijay, for excellent talk. And I must say, you are a very good teacher. Your presentation was just like a very classroom, like We feel like you are a student.
Thank you so much.
Thanks for excellent presentation.
Yeah.
We started with the basics of the nanoparticles and end up with its end results and end application. That was wonderful. We discussed about the smart targeted drug delivery system, and in reference to agriculture, particularly, you presented your lab work in terms of production, protection, preservation, and processing. So it will be a big opportunity for us to collaborate with you in terms of many aspects. Hope
Definitely.
you will allow.
Definitely. I told you.
One more
I
thing,
look forward for Jambulkar, Dr. Jambulkar.
Yes. We also like your how to prevent the pesticide loss. Yes, by making it nano stickers. And with of graphene oxide. So it's wonderful.
Thank you so much.
So I will not take much time. I will. This session is open for discussion. Anybody is having some question can ask. Please. The chat box, there is one question. Nanoparticle works as a photo protection in plants. Application of nanoparticle, we have to go through seed priming or foliar spray.
Yeah, see, photo protection, that question, very interesting. Okay, see, particles have different property. Okay, the light falls on an object, it goes into 4 phenomena. Okay, one is reflection, other one is transmission. Okay, and the other one is conversion, what is it? Scattering. Okay, one is absorption, other one is transmission, other one is scattering, other one is reflection. Okay, so these 4 are the basic phenomena. Okay, when something is going for absorption, the thermal behavior happens, photothermal happens. Okay, when something is going for scattering, okay, that particle will be good for imaging. Okay, that is how the particle, the different particle is used for different purpose. Okay, and with reference to the one that's the audience have asked, how protection is happening. Okay, photo protection. So here, I think Dr. Ramesh Alia, yesterday's talk, he was mentioning, okay, it is hard to control the particle synthesis with definite property. In case of TiO2, he was mentioning to control the synthesis, whether it should be rutile or anatase or different thing. Okay, say for example, the TiO2 is in our talc, powder talc. Okay, there it is, I think it is in a rutile phase, which is an inert phase. Okay, so it don't take the light. Okay, so when you apply the talc, it protects your body. Okay, the other one is anatase. Okay, that can take the light and it have a very active semiconducting gap, so it converts it into reactive oxygen. Okay, so if somebody applies that will completely scorch the skin and you will get cancer. Okay, so these are all very closely related phenomena. So accordingly, we have to take, and as like the audience have asked, the material like the specific phase can avoid the light degrading the pesticide. Okay, and that was the first question. I'm sorry, the second question I missed. You were saying
fungicide available in nano form.
Fungicide available in the nano form. Yeah, it is available. Yeah. I think you may have listened to many of the talks.
For further questions, you may contact Dr. Vijay Kumar in his mail.
Thank you.
Thank you, Dr. Vijay. I welcome our Honorable Vice Chancellor, sir. Also, he has joined. I request our Honorable Vice Chancellor, sir, to felicitate our speaker, Dr. Vijay Kumar, with a token of memento seminar. Dr. Vijay, please unshare.
Yeah. So unshare, right? Unshare how, I'm sorry, stop sharing. Yeah, okay. Yeah.
Yes, sir.
Thank you.
Thanks so much.
Thank you. Now I request our Director Education, Anil Kumar sir, to brief up about the proceedings, today's proceeding of the session.
Thank you, Dr. Prashant Jambulkar and other organizing committee member. Indeed, the 2 days national webinar on nano size big impact, nano revolution for transforming agriculture, food nutrition and health was held. And in this webinar, total 120 participants are there, and they are of 20 different states, and they have linked either through the Zoom as well as also through the YouTube. And today we have seen that more than 120 they are there onto the YouTube, because this YouTube has been shared to other people as well. And indeed, I am very happy today to see that the success of this webinar, because we are thinking that in the newly developing university, we have to have, first of all, the capacity building. We have to have sensitization program, so that the scientists of our university, as well as the scientists of neighboring institution, they get sensitized about the role of nanotechnology in the field of agriculture. And I think the purpose is very much solved. And in this webinar, we have total 12 learned speakers, and they all have shared the blend of knowledge in the hardcore as well as the application oriented knowledge to all the participants. And yesterday there was 3 speakers, and today we have 4 speakers. And in the first speaker of the day, Dr. Jagat Rakesh Kambar, who hails from the AIMS Bhopal, he discussed about the function of 3 molecules, lactoferrin survivin and vascular endothelial growth factor, and indeed how these proteins could be utilized for various applications in the field of medicine. So his expertise on nanomedicine has been shared to the participant, and how nicely he has covered that instead of the targeted delivery, we should also use the aptamers rather than the antibody itself, because antibody sometimes they are forming immune complexes and they are eliciting the hypersensitivity reactions as well as also the autoimmunity. So instead of antibody mediated targeted delivery, we have used the RNA and DNA aptamers for that purposes. And he also cited oral administration of the drug with nanocarrier delivered system in cancer stem cell, and also pointed out very important aspect, that is nanotoxicity, and also the various model system which could be used for studying the nanotoxicology, particularly while we are developing these nano delivery system. He also shared about the use of the fluidics devices and how they could be utilized for detection of the cancer stem cell, because cancer stem cell is generally they found resistance, and they are not able to because many times cancer drug has been given, but this cancer drug is been excluded from the cell, and hence before they become resistant. So it is very essential to find it out the role of cancer stem cell and how cancer stem cell could be targeted using these nano medicines. And then the second lecture has been delivered by Dr. Vinod Saharan on to the another very important aspect, that is of biomaterial based nano innovation for plant protection and growth. This is the second title. And in his excellent talk, he discussed about the nanomaterial for plant protection and growth promotion. He discussed about the bio-inspired approaches to develop the biogenic nanoparticle, and also mentioned about the role of the solid matrix like chitosan. And not only the chitosan, along with the chitosan, he have also used many minerals like copper, zinc, silicate, iron, essential oils, and plant glycosides, so that ultimately we can able to develop the stable nano materials or nano devices, so that they could be utilized for plant protection purposes. He also discussed about the use of chitosan as biogenic material, and because chitosan is basically showing different kind of the property until unless we are not making it soluble. So this could not be utilized as a good material for delivery of the many of the pesticide or fungicide. He also used these nanomaterials for seed treatment and foliar application against various crop disease. And he also developed the smart way of delivery system on the basis of demand and also on the basis of command. So basically, it is the material which is providing the regulation as such. So whenever the plant protection molecules is required, then they will be delivered as on the demand and also on the basis of their command. He also discussed about the role of salicylic acid and functionalized chitosan nanoparticle for disease management, and also slow and sustained release of the salicylic acid to the plant, so that they are able to elicit the systemic acquired immune responses, and ultimately they are providing the immunity or the defensive mechanism against many of the plant pathogens. He has also discussed about the biostimulant and fertilizer, that is functionalized fertilizer, and that is under trial, and he has got the basically DBT funding on this aspect, so that he could able to evaluate it under large scale trial. And third speaker, Dr. Ashok Kumar Nadda, who also developed so many of the nanomaterial and its use for the reduction of carbon dioxide. And we know very well that carbon dioxide accumulation as well as the greenhouse emission, they are causing the global warming, and climate change is also because of that reason. So until unless we are not able to sequester this high amount of the carbon dioxide available in the nature, certainly the problem will remain there. So nanotechnology also going to play very, very significant role of using the nanomaterial along with the immobilized enzyme onto the nanomaterial. They can cope up very well this carbon dioxide be emitted in the environment, so that this emitted carbon dioxide can be converted into the other useful compound like calcium carbonate, or maybe directly it is converted into the methanol, or so that it could be utilized as biofuel, etc. Fourth learned speaker, he is a very good teacher and very nicely covered the role of nanoparticle, basics of nanoparticle, and discussed about the smart targeted drug delivery system. And in reference to agriculture, he presented his lab work in terms of how these nanomaterial could be useful for production of the plant, and also the protection of the plant, then preservation and processing. And the another very, very important thing he was discussing, targeted pesticide application utilizing the graphene as a nanomaterial, then preservatives, and then also energy transfer to the photosynthesis, and ultimately how photosynthesis could be enhanced, so that we are able to get the productivity enhancement in the field of agriculture. One of the most important aspect he has covered about the nano sticker, so that these nano sticker, they can able to bind and they can overcome the drift loss. So this is the phenomena he has developed utilizing the nano sticker, and ultimately this will able to reduce the pesticide consumption, because pesticide loss is very much there. Generally, it wiped out whenever rainy season is there. So if we are using such kind of nano sticker, then they will able to persist these pesticide quite common onto the crop, and subsequently we can avoid these pesticide losses. And he also discussed about the pheromone release. So all these lectures are really scintillating. I think not only to me, my knowledge has been enriched, and beside, I think all the participants, they will be highly benefited. And I'm sure that such kind of the capacity building program through national webinar, because COVID has given opportunity that how these communication system, the virtual system, that could be utilized in enriching our knowledge. And I'm really today feel satisfied that how we can utilize our time for our enhancement of learning and knowledge, so that it could be applied in the enhancement of the production and productivity in the field of agriculture. And not only agriculture, that we have to utilize such knowledge for food and nutrition sector, as well as also in the medical sciences, so that good kind of delivery system will be developed, which are having less side effect, and also they will economize the dose, so that the cost will also be reduced. Thank you very much for giving me this opportunity for concluding. Not only today, but yesterday we also concluded about the presentation made by these 7 learned speakers. Thank you.
Thank you, sir, for your concluding remarks. Now we came to a fruitful, enlightening, and so much to take away. Before concluding remarks of our Honorable Vice Chancellor, sir, we are happy to inform you all that Dr. Tanuj Mishra and Dr. Shrendra Kumar, our colleagues at RLBCU, developed a R package for leaf area determination, visual images of the plant. They have developed R package, namely large, for leaf area determination from visual images of the plant. With this package, leaf area can be determined by only capturing images of the leaf. We know that the measurement of leaf area is important in the studies of plant biological characteristics. Leaf area determination using the currently available instruments such as LIGO, SPAD involve too much code. In this context, the package developed here is freely available in the CRAN repository. We are happy to share that within 4 days of its publishing, it has been downloaded 65 times around the world. I request Honorable Vice Chancellor Sir to inaugurate the first copyright of R package from RLBCU Jhansi in today's platform.
You are mute, sir. Jamunkar, sir.
Thank you. Now I invite our Honorable Vice Chancellor Sir for his words of blessing.
Thank you very much. I think it has been a very good webinar for about 1.5 days, and we have including the message and overall view by Honorable DDG Crop Science, Dr. T.R. Sarmadi, very learned speakers. And I am sure that the participants must have benefited out of the presentations made through webinar. We know that because of the limitations of physical presence of different participants, we couldn't organize it on campus. But in future, we plan to organize many such activities so that the interaction is worth and interaction is meaningful. Although through webinar we are able to interact, but it has no substitute of the physical presence of our learned speakers and their interaction with different faculty members and students. We are calling our students on 10th of October, PGA students, and PGA students really gets maximum benefit out of such presentations. And I'm sure that maybe in next 2-3 months, we have planned certain more activities because along with the various programs, celebrations of different days, celebration of Atal Jai Vigyan series, and many webinars in different disciplines, our faculty and students are participating in different programs organized by different organizations. The overall impact of these lectures is visible by presence of more than 100 delegates. Although we kept a nominal fee, registration fee, and the purpose of keeping the registration fee, nominal registration fee, has been so that the participants are sincere in attending. Otherwise, many times they simply register and attend here and there and then forget about it. But I am very happy that most of the participants have been very active. And those who couldn't participate can take the advantage to YouTube. The lectures would be available. And I would also request Director Education compile the lecture notes along with the overall overview given by PDG, so that we can publish it in the form of a outcome of this webinar and get it released in our forthcoming conversation. So that should be our priority, and let us see that within next 1 month time, we should try to obtain the write-up from different speakers, and after proper editing, we can publish it and bring it in a small booklet so that it gives an valuable information to different stakeholders. The role of nanotechnology, we have been hearing for 2 days, and particularly in agriculture. As I talked about, it has not only relevance in agriculture, but also in value addition, food safety standards, the medicine, and many other areas, particularly health, has got a lot of scope. And we have to explore the possibility, because being an agricultural university, we can only, intensify our research efforts, which promotes the resource use efficiency in agriculture, and also in collaboration with other key scientists can develop the methodology and also a strategy to overcome some of the issues in food and nutrition and also in health. So I am sure that our scientists would be able to draw some projects in future, submit it to the relevant organization, and try to intensify their research efforts in this important aspect. And as I mentioned yesterday, the research work on nanotechnology is there. It's not that nothing is being done. A lot of work has been done. But we need to intensify and we need to develop tangible products so that it can be commercialized. As it's one of the happiest situations, we all know that nano urea is being commercialized, and perhaps it is the first product in nanotechnology which is used commercially in agriculture and reducing the cost, quantity, and also the effectivity. As I was listening, it has got 50% saving in the quantity, and also efficiency is enhanced. So that way, we need to look at what are the other sources and what are the other areas where we can work in that direction, including its impact of nanotechnology on soil microbes, how it helps to develop, and how it have adverse impact, if any, and how we can mitigate that adverse impact, and also how we can educate and create awareness about the use of this nanotechnology and its product. Because many times people are not much aware about this nanotechnology and its application, and therefore many products, when it is said that it's a nanotechnology based product, so people sometimes hesitate to commercialize it, use it. But when the advantages are known, advantages are many, then certainly this can be very well used and commercialized. In India, there is a lot of scope of nanotechnology because we have, resource as a basic constant, and not only in effectiveness of fertilizer, but also in herbicides. We talk about the nanomolecules, where we can, with small quantities, have large effective application, better control of different pests, and even plant protection and disease and pest control. So these are the various areas through which we can have our research projects in the future. Although we have limited staff in this field, but certainly we can collaborate with the other scientists, not only of the neighboring two institutes, but also the other neighboring institutions located, in Budelkhand and adjoining areas, so that we can get the benefit. Because science has got no boundaries. We can develop projects taking the help of national and international renowned scientists. And I am sure that many new speakers, many experienced speakers have given their opinion about this nanotechnology, and the delegates participating in this webinar certainly benefited, and they will certainly take a clue from these presentations, and in their respective fields certainly try to develop some projects, and in case we can collaborate with them, we shall be happy. So at the end, I thank you one and all for sparing your valuable time. The speakers make the presentation and prepare it. Takes some time, but you have taken pains to prepare your presentation and make it available to different delegates. And also, I request you to provide your write-up within a month's time so that we can publish it. Certainly send you a copy, hard copy. Although we will put on website, but a hard copy is always released in the library so that more people are benefited and join it and listen to talk have been organized. Thank you very much. Thank you, Dr. Jambolkar, and thank you, Dr. Rani. Key person, RLBCU and IGFRI, who have given their valuable time and organized this important webinar. And we wish that next time we meet physically and we have long deliberations so that some valuable certificates are kept by the Thank you. Thank you, Manana. Thank you.
Thank you so much, sir, for your suggestions and kind words. Now I invite Dr. Jambulkar, Organizing Secretary of this webinar, for a vote of thanks.
Thank you, Dr. Rana. Thank you all. Before going to vote of thanks, we may have forgot one little thing, that our Honorable Director, Education, sir, and our organizing Chairman Organizing Committee has also delivered a lecture, and we have not felicitated him with a invited speaker memento. I will request our Honorable Vice Chancellor, sir, to felicitate our Director Education, sir, with a memento of invited speaker.
Thank you, sir. Thank you, sir.
Thank you. At the end, we came up with a very fruitful ending of two days webinar on nanotechnology. And I must, I'm sure that all of you has lot of to take away with you with this webinar. First and foremost, I will extend my highest gratitude to our Honorable Vice Chancellor, sir, who has always been a source of inspiration and a guiding lamp for all of us. And he always encouraged us to perform better and to bring laurel to the university as well as self. I also extend our thanks to our director of our neighboring institute, IGFRI, Dr. Amresh Chandra. He is always been there to support us and our university and extend his support in all sort of manner. I express my thanks to our Director Education, Dr. Anil Kumar sir, who was the torch bearer to bring out this excellent 2 days webinar on nanotechnology. Who was the brainchild? It was the brainchild of his mind, and we have formulated under his guidance. I also thanks to our speakers, eminent speakers. I must say, all facets of the nanotechnology has come to a platform because of them. I thanks to Dr. Ramesh Relia, Dr. NGH Jayadi, Dr. Jagat Rakesh Kamarji, Dr. Vinod Saharan, Dr. Ashok Nadda, Dr. Vijay Kumar, and I express my sincere gratitude to all of them, and who has spared their valuable time and knowledge with us, and knowledge and thoughts with us, and paved way forward to many young scientists and researchers like myself and many others, that they pursue their career and do something for the country in the form of good research. Thank you all. I thanks to all the participants from almost 18 states, 18 to 19 states. There are registered 111 participants to this webinar. Many 43% were the students and 57% were the teachers and scientists from around the country. it's a very successful seminar, and I thanks to all of you, and I request you in future to visit our university in person, and we will look for good collaboration with all of you in terms of this research in the area of nanotechnology.
I think,
thank you all.
As suggested by our Honorable Vice Chancellor, I request all the speaker, if you provide your write-up along with the one-page opinion about the use of nanotechnology, that will be useful. So write-up will be there on your lecture, and the opinion, one-page opinion, that will be really good. At least we can able to understand that how nanotechnology could be beneficial in field of your interest. Thank you very much.
Thank you, sir. Thank you. Participants, your certificates we will be sending by evening, or maybe tomorrow. And feedback link will also be reached to you, and we welcome your feedback for this. Thank you.
Dr. Jambulkar and your organizing team, if we can meet, so
yes,
that it's able to see how next we can do.
Yes, sir. Yes, sir.